So and all the flowers are bracteate, and all the plants in the orchids are herbs. You will not find any tree species of orchids. Some of the species are saprophytic. Some of the species are climbers. They are vanilla and all that. So they are the wondrous creation of nature, and two significant characters that actually differentiate orchids from all other plant groups. Those characters are modification of third petal into labellum, and the fusion of the male and female reproductive organs, that is called as gynostemium. That is that fusion is from base to apex. So presence of these two phenomenon that itself differentiate orchids from any other plant groups. And of course there are many other characters. The seeds are minute without presence of endosperm, so they require external supply of nutrients during their germination. So why we should study orchids? As we all know, orchids are significant for many purposes. They are very, they are highly known for their ornamental these qualities. So they are having different combination of color, fragrance. They are good, having good shelf life, and they also are used as a cut flower, potted plants, and they are highly compatible for hybridization. Their reproductive compatibility is much more than any other plant group. Then many of the orchids are used in traditional medicine, and they also used to show mimicry for different purpose in their life cycle. And they are of course botanical curiosity. If you look at the life forms of orchids, they vary from miniature, maybe 2 to 3 centimeter size of the plant, to the several meters of climbers in vanilla, and even the bulkiest orchid of the world, that is Gametophyllum speciosum, that weighs about a few tons of kilogram. So as I said, when we look at the importance, how these orchids can be classified into three categories. They are mostly medicinal, ornamental, and botanical curiosity. If we say about medicinal value, you all know the terrestrial orchids, they possess the underground tuber. In fact, the name orchid, it is derived from a Greek word orchis, that means a pair of testicle. That actually, according to doctrine of signature, that any plant part that resembles a human organ is responsible for some treatment of that particular organ. So this underground tuber, that is pair of testicle, that is considered aphrodisiac. And yes.
Yes, sir, your
Yes,
slides are not.
they are not moving.
Yes, sir.
Is it moving or what?
No, sir. The slide is stuck to the first slide.
Oh. Stop sharing. Then window, na? This one.
Please put it in full screen mode, no?
Yes,
Open
yes.
it in full screen mode.
Yeah. It is moving now.
Start moving it. Then you can see.
Is it moving?
No.
No, sir.
Just a minute. Is it moving now?
Yes, yes.
It is moving only by the click of the cursor. It is not automatically moving.
Okay, yes. Now it is moving.
Yes, it is in full screen mode now and it is moving. Yes, good. Very nice, sir.
Should I continue now?
Yes, please.
Okay. So I was talking about the importance of orchids. They can be categorized into these three categories. And if you look at the resemblance of this underground tuber, they are considered as highly aphrodisiac. And these terrestrial orchids with such type of tubers, they are used in vitality medicines, and they are used for the growth and vigor. You see the tubers here. You see the Salam Panja, what is called the Dactyloraja hatajaria and Gymnodenia orchidis, these two species. And they are also a part of Ashtavarga. So the good number of orchid species, they are used in medicinal industry. And of course, you all know what kind of ornamental value orchids bear. They are highly compatible for hybridization, and we can play with any type of color combination, and we can create whatever we wish with combination of orchid flower and their ornamentation. And of course, there are botanical curiosity. You can see the spider orchid, moth orchid, tiger orchid. These, mimicry or these things are, that is for attracting pollinator or maybe for adaptation in their life cycle. So when we look at the past research on Indian orchids, it all started with Charak Samhita, where some of the Bendaceous orchids are mentioned in the name of Beneda in Sanskrit. So then the first, organized study on Indian orchids were done by Von Reed in that Hortus malabaricus. And there are a series of workers who had worked after the Linnean period, 1753. Then Wallich, Roxburgh, White, Lindley, Griffith, Lindley again, then Reichenbach, Drury, Atkinson, and J.D. Hooker in his Flora of British India. He has treated orchidaceae comprehensively. In the present era, the Indian orchids mostly they are revised and monographed. Some of the genera, Cymbidium, Calanthe, Silogine, all the lady slipper orchids, they are all revised and monographed by different people during past 2 decades. So we have good information on Indian orchids through this literature. Some of the checklists and enumeration what we have for Indian orchids. The first significant one was by U.C. Pradhan in 1976 and 79, 2-volume book it is. Then S.K. Jain and Mehrotra in 1984. Then S. Karthikeyan et al. In 1989. Then Satish Kumar and Manilal in 1994. KP Singh. Sorry. Is it right? I continue.
I request Pants Benny to please mute your mic. Yes, sir, you can.
Okay.
See it.
So these are the checklists and enumerations published for Indian orchids so far. And when we look at the diversity, the latest estimate is 1270 taxa under 155 genera. And you can see the 10 dominant genera with Dendrobium, Bulbophyllum, Ebeneria, Iria, Oberonia, Liparis, Silogine, Peristylus, Cymbidium, Calanthe, and Eulofia. These 10 genera together, they contribute more than 50% of Indian orchids. And rest of the less than 50% is represented by 145 genera. So they are highly, polarized in their diversity. The differentiation is happening in few genera and not in other genera. And heavy with number of taxa means this is 760 taxa. They are epiphyte mostly, and 498 taxa they are terrestrial. They include saprophytes also. When we look at the distribution pattern in India, of course, Himalayan region is, you all know, it harbors the maximum diversity. There are many factors responsible for this. Most important is the topography and the position and the climate. So 963 taxa it is reported from Himalayan region, including the Northeast India, and 371 taxa is Eastern Ghats, Western Ghats together in the peninsular India, and 125 taxa it is reported from Andaman and Nicobar Islands, and 308 taxa are endemic in India. So the orchids in broad sense, it is classified into some subfamilies, and there are many classifications available. The latest and most acceptable one is by Mark Chase et al. In 2015, So of course there are few deviations available from that classification now itself. We classify Indian orchids fit into these subfamilies. Number one is Apostatioideae. This is represented by one genus only and 3 species in India, and this is highly known for its 3 fertile anthers. So people can easily misinterpret this species as a Liliaceae or other monocot member. So they are orchid, but they don't look like orchid in their natural habitat. Then subfamily Vanilloideae, it is represented by 6 genera in India, and they are mostly this saprophytic Galliola, then Lecanorchis, and some of the these things autophytic genera, that is Vanilla and Pogonia. These things comes under this subfamily, and they also available. They are available in subtropical to temperate zone. Some of the species are found in tropical area also. Then subfamily Cypripedioideae, you all know how important they are, how costly they are. They are lady slipper orchids, and they are highly endangered for their ornamental value. These Paphiopedilums and Cypripediums. Paphiopedilum is known by 9 species in India, and Cypripedium is known by 4 species in India. So these 13 species together, they they are highly precious. They contribute to a lot of hybrids in these lady slippers. They are used as one of the parents, and this Paphiopedilum perianum, this top left figure, this was once declared as the lost orchids, and there was a prize money announced for relocating this species, and it was relocated in Arunachal Pradesh, and I have seen the habitat in that Kameng district near Dirang. Then this Tenga Valley, there is a very good population available of this species Rupa in that area. Coming to Orchidoideae, these represents the most of the terrestrial orchids like Habenaria, Peristylus, Platanthera, and they are also having a lot of beautiful flowers, but they are more important for their medicinal value, and they have certain pollination mechanisms to attract the pollinators and get the pollination happen, and they mostly inhabit the tropical region as well as the temperate, subtemperate, and even alpine region also. So this is one of the significant group of orchids. And this subfamily Epidendroideae, it is the largest one. This represents both monopodial and sympodial epiphytes mostly, and some of the terrestrial genera like Calanthe, Cymbidium, Eulopia, they belong to this group. And I will show you some of the more pictures in subsequent slides. They are again found in mostly tropical warm climate, the rainforest, and some of the species are even found in temperate and alpine zone. Then we have a challenge. Means we all consider orchid as one of the largest families of flowering plants in the world, with more than 28,000 accepted names. But when you look at the flip side, for the 28,000 accepted name, we have more than 48,000 synonyms. So of course, synonyms are there for all other plant groups, but in case of orchids, the number of synonyms are quite high. And if you look at the Indian scenario, this ratio becomes 1:5. Now here it is 1:2 in the global scenario, but in Indian scenario, the ratio becomes 1:5. So this can be attributed to many facts. Maybe we can, if we introspect our quality of research, there are many things. Means when we cannot link the extremes of species concept, the evolution of species, when we cannot link and indiscriminately we try to publish new species everywhere, then this will be the result. You can see the signature characters in orchid, identification and taxonomy. The signature characters that lies in the labellum. The labellums are unique in most of the species, and they will not match with each other. You see, this is one genus Dendrobium. You see the different type of labellums found there. So the texture, lobation, coloration, ornamentation, they all are different. So when we look at these things, we will be able to differentiate the species in a correct manner. You see, this is another genus, Iria. You see the labellums are different in all the flowers. Then when we look at the diversity in a higher hierarchy, maybe at the level of genus or at the level of tribe or subtribe, then we may have to look into the diversity in pollinia, structure of pollinia and pollinaria. Here you can see they are different, means the nature of pollinia, the presence or absence of basidiums, types, other ornamentation, they actually really matters. And they differentiate orchids into the higher groups. Another phenomenon that prevails in orchid, that is polymorphism. In a different developmental stages, orchid exhibits different, same flower, it can exhibit different morphology. So it is really important to study the flowers, the specimens at the field and in different seasons and different developmental stages. The same inflorescence, if you dissect different flowers, the lower flower will be having 2 pollinia, the immature flower is showing 4 pollinia. So 2 pollinia, 4 pollinia, even the subfamily are different. If you look at the key to the subfamily, the Orchidiode subfamily is having 2 pollinia, but here the same plant is having 4 pollinia. Likewise, you see there are some constant character and there are some variable character. When we look at the constant character, that will remain constant. Here you see the length of the mid lobe. This is always smaller than the length of the side lobe. This is one species, this 4 image, and you can see the live picture here. The length of the mid lobe is shorter than the length of the side lobe, but the variability is the length of the side lobe. The side lobe length is variable. It can vary from very small to a coil like this thing. So when we cannot understand this phenomenon of constancy and variability, we attempt to publish new species, and that result in creation of synonyms in subsequent studies. So another example of polymorphism, you see the number of callus in the labellum. You see here it is 3 callus. You see the 3 callus in the mid lobe. The same specimen, same plant in a different flower, the number of callus are 5. You see. The number of this callus again is an important character people use to delimit and describe new species. So we need to understand and study very carefully. Otherwise, again, the number of species will be amplified to many degree. Another example, you see the variation. Here, the presence or absence of this spot, it is very important character to identify a species. You see the variability, whether the spots are present in the ventral surface or dorsal surface, whether they are absent in both the surface, present in one surface, absent in one surface. They are all linkages of one species. Different populations, they used to exhibit different characters. So when we fail to understand these variation and link them, then it creates new species. Another example, you see these 2 species are considered as synonym in the world databases, but I feel they are different. I can show you the proof why they are different. You see, these are the gynostemium or column of 2 species. In one species, the rostellum is present. In another species, the rostellum is absent. And the function of rostellum is to prevent self-pollination. So in this species, it is cross-pollinated. This species, it is self-pollinated. You see the shape of the anther. Here it is semicircular. Here it is triangular apiculate. Then you can see the significant, stigma lobes, 3 stigma lobes here, and here the stigma lobes are merged in the stigmatic cavity. And the same species, when you see the pollen, apparatus, the pollination mechanism, here the pollens are attached through the middle and pre at the top and bottom. Here the pollens are pre at the middle and attached at the top and bottom. So this type of critical study is required to delimit a species or differentiate a species from each other. Again, the orchids, they exhibit post-anthesis changes also. After pollination, some flower feature they used to change. Maybe the petals are elongated. Maybe the pedicels of fruit, they are elongated. So many things happen after the pollination is over. So if we consider those things as morphological anomaly and publish them as a new species, then we are not doing justice to taxonomy either. Okay, I will give you the picture of some of the valuable ornamental genera which are establishedly used with the Cymbidium, Dendrobiums, Venda, Phalaenopsis, Paphiopedilum, Coelogine, Thayas, Iria, jewel orchids, Cypripedium. They are well used. That doesn't mean that other orchids, other species are not ornamental. They are also equally ornamental, but they are not so popular and they are not so far domesticated in that level. So we need to understand what quality every orchid is having, and we need to harvest that quality and make it into our use. Likewise, some of the medicinal genera are Eulophia, Cryptidium, Dactylorhiza, Habenaria, Gymnadenia, and Platanthera. They all possess the underground tubers, and they are highly medicinal in traditional system. Okay, I will quickly take you through the different group of orchids, the diversity, enormous diversity what orchids exhibit. They are all Indian orchids. Bulbophyllums, you see, that is the highest number of species it is having, nearly 140 species. And they are again variable in their floral structure, vegetative morphology everywhere. So this is the highest diversified orchid groups in India, Bulbophyllums. Then comes the Dendrobiums. Arunachal Pradesh is very, highly diverse in these groups. And Dendrobiums are really beautiful. You can see the color combination and all that. Then the Oberonias. We have nearly 60 species of Oberonia in India. And they are minute plants, but their floral architecture is marvelous. They used to be present in long verticillate inflorescence, but the labellum is really quite ornamental if you can see it closely. Then the Irias, I did my PhD on this group. It is having 61 taxa in India, and of course they are not so well known for the ornamental industry, but they have high potential. Even some of the species are already in use, but all the species can be used for different purpose in ornamental industry. Then the Habenarias, they are mostly terrestrial. Then the Cryptidium and Liparis, they are known for medicinal use. Cryptidium, acuminatum, then this Dienia muscifera, they are the part of Ashtavarga, and they are highly used in that Ayurveda industry, Chamanpras and all, it is prepared. Then Coelogines, you see the Coelogine barbata, stricta, then Corymbosa, Fusescens, a lot of good species we have, and they harvest fragrance. The white flowered Coelogine, they are good. We can even utilize them. Their fragrance can be utilized for many purposes. Then Thunia and Pleone, they are again a relative of Coelogine. They are also highly ornamental. Then the lady slippers, then the jewel orchids, Thunia and sorry, this is wrongly labeled. This is Kalanthi. They are a group of terrestrial orchid. Then Venda and Alice, they represent the monopodial orchid group. The growing apex is unlimited here, and this group especially is highly compatible for intra and intergeneric hybrids. So many of the blue Venda, red Venda, they all belong to this group. They are highly used in this hybridization industry. These are all photographs of this is Acampe, Phalaenopsis, Eridis, and then Rhincostylis, and this your Smithinandia. These things are there. Then again, Venda and allied groups. You see Copperpool, you must know in Northeast India, Copperpool and Bhattopool, Rhincostylis and this Papilonanthe. They are having highly cultural significance in Northeast India, especially in Assam. And this is the red Vanda, Renanthera imsutiana. This is one of the parent to many of the good hybrids. Then saprophytes, this is Galeola lindleyana. Alpine orchids, Satyrium nepalensis, Satyrium ciliatum, then the Puner orchis tusua. Okay, now we have seen the enormous diversity what we have in India, orchid diversity. In India, it is the second largest family of flowering plants. Then now the second part of my presentation, it will cover the red listing part. So what documentation we have so far for Indian orchids? Red Data Book of Indian Plants, it was published in 3 volumes and it includes 65 species of orchids. Then Iucn 97 Red List, it includes 105 species Indian orchids that belongs to 46 genera. And all the Orchidaceae are declared as threatened under Schedule 38 of Biological Diversity Act. Of course, 12 species are specifically mentioned from different states, and entire Orchidaceae is included in citation negative list of export, and even in the WPA, that is Wildlife Protection Act 1972. So these are the global and national legislation available for orchid conservation and utilization. I will not go in detail because I have to take time to explain the Iucn procedures. So these are the global and national legislations. So I must say why we should do the red listing. We have to find out why it is required. When all the orchids are already in different legislation, it is conserved by default. Then why should we do the red listing? But I must say there are many species they do not qualify for being called as a threatened species because of their non-significant use and maybe their more wide distribution and good population size. So unless and until we utilize our biodiversity to an optimum level, biodiversity has no value. So that's why we must evaluate the orchids, particularly to find out which species should be qualified and which species should not be qualified to be called as a threatened species. So the widely accepted system is Iucn and red listing assessment system. So for this purpose, we are taking Orchidaceae as one of the candidate group for this Iucn red listing. We have completed a project on this. So to introduce the Iucn red list to the participants, I will start with the definition. What is Iucn red list? It is not just a list, but a compilation of the conservation status of species at the global level. And this is based on the best scientific data available. And it is globally used, and it influences the biodiversity conservation mechanism. And it provides information analysis on the status and trends to control the rapid loss of a particular species. Then it is a process. It starts with a data collection, then the expert workshop, then reviewing, and then quality control. Then how the red list data can be used. Once we create the data, we must know how it can be used. This can be used for analysis and information, then conservation planning and priority setting, and it again can be used for international conservation policy, inform the private sector decision making, education and public awareness. And it gives us the idea of how is the likelihood of a species becoming extinct in the near future, given the current knowledge about population trends, range, and recent or current projected threats. If we know today what is happening with a species, we can easily apply the Iucn system and project what is going to happen in the future also, if the today's activities are not controlled. So we must know the components of a red list assessment. It used to be divided into 3 broad category components. One is the red list category and criteria. The number 2 is documentation supporting the category and criteria. We must be able to justify if we are saying it is critically endangered, why it is critically endangered. We have to support this statement with proper documentation. Then the most important aspect is distribution map of that particular species, what we are assessing. So these are the 3 components. Then what can be assessed? we can assess all the described taxa at the level of species or below. We cannot assess a genus because genus is a one entity that actually represents multiple taxa. So a species, which is the smallest unit, species or any rank below it, means variety, subspecies or forma, that we can assess. And of course, we can also assess the undescribed taxa, that is, if it is a clearly distinct species, and if we have the museum or herbarium voucher reference provided, and if we have the distribution information available, and there is a clear conservation benefit for assessing an unknown species. So these are the things we can assess, and we can assess the things, species, taxa at the global level. At the same time, we can assess the species at the regional and national level also. And we always assess the wild populations. We do not assess, IUCN will not support assessment of introduced population or maybe in cultivated state. So wild population in their natural range, they are qualified for assessment. So what are the tools for red list assessment? This is the IUCN red list categories and criteria. All the categories and criteria, they are defined in this thing. It is available in that IUCN website. PDF version is available. Then this is the guideline for application of IUCN red list criteria at regional and national level. And this is again the guideline in a broadly elaborated manner. And this is the summary sheet. You can see criteria A, B, C, D, and E. All the thresholds are mentioned here. So we have to collect information and analyze the information based on these thresholds. Then we will be able to assess the threat status. Then this is the document which can be used for consistency check. And finally, we get everything in this website, iucnredlist.org. Here we will get all these PDFs and all the procedures. Even we can train ourselves, sit for the exams, and we can be considered. We can get the certificate of red list assessors and trainers and everything. We can get it from here. So it is a process, as I said. First is pre-assessment. We have to collect data and compile it into the IUCN format, the datasheet format. Then we have to assess the species independently or at a workshop, assign the red list category and criteria, then review and consistency check. It can again be done in the workshop or at the individual level, and then it can be submitted to the IUCN site. That will be finally reviewed by the red list unit of IUCN, and it will be published in the red list. So this is a process. This is the process of a red list assessment. Once it is published, every species will be having a separate DOI. So it will be very useful for the career also. Number of publication and all that will be very significant. So what is the role of a red list assessor? The red list assessor will compile all the currently available data on population status, distribution, ecology, use and trade, threats and conservation measures across the species global range. And data may come from published literature, personal knowledge, and even grey literature on published report also. And the assessor will have to assign the red list category and criteria based on the available information and justify the assessment following the documentation requirements. And map is also required to be prepared and submit it to the for the review at the IUCN red list unit. So these things are a matter of separate lecture. Actually, this IUCN red listing, it requires a 5-day workshop to be properly understood. But still, I will venture into making you aware about some of the definition. This is population. This is highly used in IUCN red list assessment. What is a population? This is number of matured individual, or the total number of individuals present across its geographic range. This is population. And population size is number of matured individuals in across its geographical range. So we must not confuse population with population size. Then subpopulation. All the individual patch is called as subpopulation. Then generation length, it is again used in different criteria, especially in criteria A and C, and the average age of parents in the current cohort, that is called as this. It can be calculated in this way: number of offspring, and the age. You can see age at first breeding and age at last breeding, and the mean age. This is the generation length. This is useful. This is reduction. Reduction is a decline in population size, at least the percentage stated in the criteria A. Then continuing decline. These are all need to be understood before we venture into IUCN rate listing. Extreme fluctuation. This is one species is found, in a big number at one location in a particular season, and the species is disappearing in a particular another season. This is usually, applied for migratory birds and all. Then severely fragmented. This is when a species is distributed in the area, dissected by unsuitable habitats, and they are highly, distant, where the possible gene flow between the subpopulation is very less. This is severely fragmented, and when more than 50% of population found in small and isolated subpopulation, it is called as severely fragmented. You see, this is the level of isolation. Then this is most important: extent of occurrence and area of occupancy. Extent of occurrence is we have to draw a polygon connecting the outermost boundary of all the known localities and all the areas, whether it is suitable habitat or not, whether it is occupied by the species or not, they will be falling under that extent of occurrence. This is the extent to which the species occurs. And the area of occupancy, this is the actual area occupied by the species. This is, you have to create a grid of 2 by 2 kilometer. This is the thumb rule of IUCN: 2 kilometer by 2 kilometer. We can even increase or decrease the grid size also, and we have to count how many number of this square is occupied by the species, and then we find out the area of occupancy. So these two, EOO and AOO, they are highly, extremely essential for this rate list assessments in under criteria B. Then location. You see, location may not be confused with subpopulation. This species is found here. This is one subpopulation. This is another subpopulation. This is another subpopulation, and this is another subpopulation. But location is such a thing where one threatening event can create danger to the existence of that particular species in the entire area. So those entire area will be called as one location if it is endangered by one threatening event. I will give one example of this river ecosystem, and this is a lake ecosystem. The same species is found in river, and the same species is found in lake. Then one threatening event coming offsite in the river, that invasive species comes, and it impacts the entire river ecosystem because there is no barrier. River flows, and the cause of this threat also flows. And the same, this is one location. This river is one location, and this lake is not affected by the invasive species because it is embedded in the river. So this area, in spite of having 1, 2, 3, 4 subpopulation, we can consider them as having two locations. Then quantitative analysis. Here it is if we do a specific assessment by doing a population viability analysis over the period of 10 years or 3 generations, then we can be able to do the quantitative analysis. This is criteria 5 of IUCN. Then the IUCN categories. I'm talking about the categories since quite few minutes now. You see, there are 9 categories. One is not evaluated, rest all are evaluated. Then the data deficient, adequate data, then the least concern, rare threatened. Then these 3 are the threatened category: critically endangered, endangered, and vulnerable. They are the threatened category. And extinct in the wild and extinct. These are the 9 IUCN category. Any species, any described species in this world can be assessed into one of these 9 categories with the currently available data. We don't have to collect any data. What data is available, with that data itself, we can assess to any of these categories. So the extinction risk is lower to higher. From least concern to extinct, the risk is higher. So the red list category used to change. There are various reasons for a species to change the category. The two regions are, one is non-genuine and the other one is genuine. When we look at the non-genuine region, today we have assessed a species as least concern. And tomorrow we are getting some new information and that can be qualified as, not even this near threatened or even the threatened category. So taxonomic changes also will happen and it will change the species assessment category. Incorrect data used previously, then criteria revision and the knowledge of the criteria. These are all non-genuine change, and we have to again look for the genuine status change. We have to look for the genuine status change. What is genuine status change? Genuine status change is, today one species is critically endangered. We have imparted some conservation measures and the species has recovered well, and after 10 years when we again reassess it, the critically endangered become endangered or vulnerable. So that is truly it has improved or truly it is degraded. So that is genuine status change. So red list criteria, nature of the criteria, you see criteria A is population reduction, B is restricted geographic range, then C is small population size in decline. Criteria D is very small and restricted population, and E is quantitative analysis. So we have to fit our collected information, whatever information on the species we have to fit our information onto these formulas and find out what our species is. So we can use the multiple criteria also. If our species is under criteria A, it is critically endangered, and under criteria B, it is endangered, then we have to take, it is critically endangered. The highest category of threat, we have to agree. So why use multiple criteria? I have said all taxa being assessed must be evaluated against each criterion, and meeting any one of the criteria qualifies a taxon for listing at that level of threat, and all criteria met at the highest level of threat should be listed. So what I said, that critically endangered is the highest. So if by applying all the 5 criteria, if we are assessing our species as critically endangered in one criteria, then we have to take the species as critically endangered. So this is the summary sheet in detail. You can see the criteria A. Criteria A, again, the thresholds are critically endangered, and vulnerable. There are the percentage of population reduction and all these things are defined. And the sub criteria A1, A2, A3, A4, then the Roman, this alphabets, this A, B, C, D, and E. These are all sub criteria. We have to evaluate our available information and fit into these things and select what actually sub criteria is fitting to our available information. Then we will assess the species. So this is the summary sheet. This is most important. So these are the 5 criteria. These are the mathematical threshold. Criteria A in detail, you'll see the population size reduction, measured over longer of 3 years or 10 generations. We have to see the generation length here comes into play. If we know the generation length, we will know the 3 generation. And if 3 generation is more than 10 years, then we have to take that time. If 3 generations it is less than 10 years, then we have to take 10 years as the assessment period. So there are sub criteria A1, A2, A3, A4. And if the percentage of reduction is more than 90%, it is critically endangered. If it is more than 70%, it is endangered. If it is more than 50%, it is vulnerable. So we have to very judiciously apply all these things, our own observation, all the literature data, and even the local community people's observation. We have to find out, and we have to select the criteria. These are all, you see, criteria B. Criteria B is all about geographic range in the form of either B1, that is extent of occurrence, and B2, that is area of occupancy. We can individually apply B1 and B2, and we can together also apply B1 and B2. So extent of occurrence is, if it is less than 100 square kilometer, it is critically endangered. If it is less than 5,000 square kilometer, it is endangered. If it is less than 20,000 square kilometer, it is vulnerable. Then area of occupancy, if it is less than 10, it is critically endangered. Less than 500, it is endangered, and less than 2000, it is vulnerable. And along with this, it has to meet any two of these three sub-criteria. Severely fragmented or number of location, then continuing decline and extreme fluctuation. It has to meet, if it is meeting this alone and it is not having any of these three, then it can be assessed as least concern or near threatened. Then criteria C, again, it is small population size and decline. If you go through in detail, you will get to know the percentage of reduction. Again, the number of matured individual will come into play, and over one generation or two generation or three generation, again generation length is coming into play. So you need to collect and analyze your information accordingly. So this is all about criteria C. Criteria D, it is very small or restricted population. Many species we used to provide a statement that we could able to see only 10 individuals. We could able to see only 20 individuals. There is nothing more left. So as long as nobody else is seeing 100 individual or nobody else is seeing more number of individual apart from what you have mentioned, so that can be taken as the number of individuals known for that particular species. So criteria D can be applied if the number of individuals are less than 50, it is critically endangered, irrespective of any other factor. If the number of individual is 250, less than 250, it is endangered, and if it is less than 1000, it is vulnerable. And D2 is again, it is a bit complicated with many aquatic species. They used to migrate from one place to another place. So the AOO is usually less than 20 square kilometer, or number of location is less than 5, and D2 only applies to the vulnerable category. D2 cannot be applied to critically endangered and endangered. So just to find out D1 and D2, we have to have some more information required. Then criteria E is all about quantitative analysis, that is population viability analysis of a particular species. Then the importance of regional red list. Regional red list, they monitor status of biodiversity in the region, identify priority species and habitats, identify knowledge gaps, and they assist in site-based conservation planning, communication and awareness raising, provide species input for environmental impact assessment, and help guide allocation of resources for biodiversity conservation. And they also support policy development. So we must have a regional red list assessment done for our own native species so that we can catalyze the conservation action. So the regional versus global. Iucn red list, what is the tool for regional assessment? This is the version 4 of this guideline for application of Iucn red list criteria at regional and national level. So this can be used at regional and national level, and it can be freely downloadable from the Iucn website. So the regional versus global red list, you see the species is distributed globally in many countries, and it is least concern. Now, if you look at the species is narrowed, globally it is least concern, and this particular country, it is threatened, because this is found only in a small population, although it is found globally in very good number. Then here it is again, you see, globally threatened because the total number of these locations and this spread is very limited. But regionally, if you see in this country itself, it is having very good distribution and very good population size. So it is regionally least concerned and globally threatened. So in this way, it has to be interpreted. And the categories at the regional level, it is having 2 more extra category in addition to the 9 at the global level. At the regional level, we have 11. These are the 9 categories at the global level. And the regional level, we have 1 more category that is not applicable, and another category that is regionally extinct. So what are the taxa? Those are not applicable. The non-native taxa, because as I said in the first, that we have to assess only the wild population in the natural range. But exotic species or introduced species which are naturalized and they do not belong to our country, we are not required to assess the threat status. That is, they are not applicable for a region. And the other additional category, that is regionally extinct, that is applied when a species is found at the global level, but regionally it is extinct from that particular country or region. So that can be, additionally added, that one more category, that is regionally extinct. Then regional assessment is a three-step process. Number one is we have to identify the not applicable taxa and decide which taxa we have to assess. Then number two, second step is to assess the applicable taxa as per the global assessment tool, and finally complete the regional assessment with looking at that potential rescue effect from the other countries. If a species is found in Sikkim and Bhutan, and we are assessing it for India, then we have to see if it is threatened in Sikkim, but it can be rescued from Bhutan population. So we need to downgrade or upgrade our assessments looking into these aspects. Means whether the population or gene flow can be cross-border gene flow can happen, or it is severely fragmented across the border also. So this is three-step process. So how we can document the red list? Red list documentation, it requires a very well-defined data. It requires whatever we say, we have to prove it through the data. So the documents, that data sheet, it is having a format. We collect the data in these formats: taxonomy, common names, red list category and criteria, countries of occurrence, map of distribution, rationale for assessment, habitat preferences, major threats, conservation measures, citation list, regions for any category change, name of the assessor. So these information we have to compile and keep it before we start the assessment process. So I must say taxonomy is a complicated world. I will quote you some examples, because all the plants and animals, they are governed by these two nomenclatural code, that is International Code of Zoological Nomenclature and International Code for Nomenclature of Algae, Fungi and Plants. If it is not according to these codes, we may end up assessing the same plant under different names. Then you see here, it is a bag of apple. All these 7 individuals are considered as apple, and the same 7 individuals by another taxonomist, the apple is only one. All rest of the 6 belong to some different plants. So these actually have a very big impact on red list assessment. When we consider all these 7 as apple, our assessment will be different. When we consider only one as apple, our assessment will be different. So you see here the apple is least concern, and here the apple is endangered. Although the individuals are same, it is only the perception. If we perceive everything as apple, it is least concern. If we perceive everything as different species, only one as apple, it is endangered. So taxonomy has a major role to play in this red list assessment process. Then if we look at the Indian orchids, as I said in my previous segment, Indian orchids are more harassed with this number of synonyms and all this. And again, in Indian scenario, we are constantly discovering new taxa and new distributional records, and we have been confused with the global classification based on molecular studies and this, that. Nowadays, every second day you will get a literature. They will merge your species, or they will transfer your species to some other genera, and you will end up at doing a wrong red list assessment. So what challenges we face in applying criteria A of Iucn? Because if we look back, this Iucn red listing system and this has come into place in recent times only. Previously, there was no such assessments done in a regional level or at the global level by with such a evolved, well accepted standard. So we have very little or no data available on population size of Indian orchids. We have, barring a few sporadic examples, all the species are not counted, or means population data is not available on all the species, then it is very difficult to apply criteria A. Then we means don't have the data on past population size, and even the present population size also we cannot determine of all the species. It is a time consuming process, and it requires a lot of fund. So these are the means challenges we have for applying criteria A of IUCN. Then criteria B, it is widely used, and what data we have so far, we can fit into criteria B, but again it requires a lot of time and resources, because we have to map all the species, and before 2 or 3 decades there was no GPS available, so all the old herbarium specimens, they are not geotagged. So unless and until we have the latitude and longitude data, we can't plot a map. So all the old herbarium specimens, we have to find out their geotag tentatively by visiting in the Google Earth or any other domain, and assign a lat long data geocoordinate. Then we have to put it into the Excel sheet, and finally we can plot the map. So we have more than 30,000 herbarium specimens which are available for Indian orchids. So this is again a time consuming process, but it can be done. It can be done, and it is being done by BSI. And BSI holds the highest number of herbarium holdings for all the plants in India. So this is the reason why BSI should be the indispensable part in any red list assessment project carried out in Indian context. So without BSI's data, it is means hardly possible, or maybe not possible to assess a species correctly, because BSI holds the herbarium holding in past 200 years, what we have done the research in India. Then criteria C, D, and E, again, these things require the population size and past population decline and present population size again. Even D also, if we say today I am going to a field, I can see Gudira repens, I can see 50 individuals there. I can say, oh yeah, 50 individuals are found. But the same Gudira repens anywhere else, it may be found in thousands. So I cannot say authoritatively that only 50 individuals are found. So this way, these are the challenges what we have for assessing this red list assessment of Indian orchids. Then few examples I will put. This is Iucn Categories. This was found in Andaman and Nicobar Island, and later on it was assessed as critically endangered by one of my colleague, Dr. Abhishek. And the extent of occurrence here is less than 100 square kilometer. It is found in only one locality, and area of occupancy is less than 10 kilometers square. It is again found in one locality. Number of location is 1, and projected decline in area, extent, and quality of habitat, that is subcriterion B3. So based on these information, this can be assessed as critically endangered B1, B2, A, B3. So this statement itself, this indicates this CR B1, B2, A, B3, this indicates the justification of how and why this is assessed as critically endangered. So these are the threats listed for this particular species. This is the example of endemic species. And this is another example. This is non-endemic species. This is found in that Kedarnath area of Western Himalaya. Only one location that time, but now it has been recorded from Eastern Himalaya as well, Neora Valley and Darjeeling area, it has been recorded. So now initially we have assessed this as critically endangered, but now we have more information. We can, if we reassess it now, it can qualify for endangered. So again, the same thing, critically endangered B1, B2, A, B3, and D. So this is the plant, this is the map of the species, and this way the red listing can be done. These are some more examples. You see Acampe pepilosa, Acampe ocracea, Acampe rigida, Acampe pepilosa variety flower. This is endemic. These three are non-endemic. You can see here the maps. You see the polygon. This polygon, all the area coming inside the polygon is EOO, and the spots, what you can see red spots. You can count the number of grids here, and you can find out the AOO. And then you can do the assessments. It is vulnerable, it is endangered, it is again endangered, and it is critically endangered. Some more maps we have created. You see, nowadays there are many domains available for creation of map. Pre-domains are available. This is GeoCat map. If you have the coordinates, that lat long data, you can easily plot this map, and that map itself will count the area. If this polygon is 555, this 5,55,872 square kilometer, this polygon. This is counted by the calculated by the system itself. So the system is very easy. Only you have to do is you have to collect the coordinate points. You see another species. This is the polygon. Another species. This is known from only one location. This is again known from only one location. So the EO, AO both are 4 kilometer here. 2 into 2 is 4. So this is a data of completed project. I have completed a project on red list assessment of Eastern Himalayan orchids. 770 taxa we have assessed for India, and you can see the vulnerable outnumbers the all other category. 505 taxa are vulnerable, 108 is endangered, and 74 are critically endangered. Near threatened 50, and least concern 18, and data deficient 15. Why they are data deficient? They are ambiguous in their identity and distribution. Then what we look forward, being in the BSI, being in the apex body of Government of India for survey and exploration of Indian flora, we have done the species inventory and taxonomy. We have completed the threat status assessment of majority of orchid species, and we are looking for doing it for the rest of the endemic and non-endemics of Indian flora. And germplasm of nearly 50% of native orchid species we are maintaining in all our campus gardens and 2 national orchid area. And we have also facilities of in vitro propagation and molecular characterization. So we plan to complete the threat assessment of remaining species, and we do the morphomolecular characterization and coordinate the R&D on Indian orchids. And we also look forward to popularize, educate, and demonstrate the cultivation of commercially important species, and explore the wild relatives or native species for commercialization, and contribute for ex situ and in situ conservation of country's important genetic resource. So thank you very much. Thank you once again for giving me opportunity to present my research in front of all of you. Thank you.
Thank you, Denise, sir.
Welcome.
Now, we can take few questions from the audience. If someone have any queries, you can put forward. Anyone? Anyone? I guess there is no one.
Online participants.
Okay, questions from online participants from headquarter itself. Yes, there is one question from student side. Yes.
Good morning, sir.
Yeah, good morning.
So you have given us a great length of information on threatened orchid species. But sir, I would like to know if there are any data regarding the declining numbers of particular species of orchids in Arunachal Pradesh.
It's a good question. There is no data available as such because Arunachal Pradesh is having the highest orchid diversity in India. It is having more than 600 species, but any particular species has to be counted in the field, and the population size has to be recorded. But this work has not been done on this aspect. So this can be taken as a challenge by the students of Arunachal Pradesh. So you all have the list, you all have the identity, photographs of all those orchid species, you know the localities. Even I visited Arunachal Pradesh for quite a good number of times, 8 times I visited. I have done my field work in Lohit, Anjao, Subansiri, Kameng, Dibang Valley, and even Siang, that Pasighat, Jenging, Tuting, this area I all visited. But no information as such on what you are asking, that a percentage of population decline or population size itself we do not know. We can estimate it is maybe 500 or maybe 1000 individual, but we cannot say exact number.
Thank you, sir.
Thank you.
That's how it was very nice and excellent presentation. So offline, online candidates, they have learned more. So very beautiful slides you have shown. That's a informative lecture. Thank you.
Thank you.
Uh,
Thank you.
Okay. We have a very special number for our viewers. Actually, students from Deranatun Government College who is attending the program from our auditorium. Some of the students wants to present their songs, their talent here with us. So without further ado, I welcome Miss Nyanching Sang Sakrinso to present a local song.
इसके बाद डॉक्टर गोपाल कृष्ण जी का प्रेजेंटेशन है.
My name is Nancy Sakumo from DNG College Founder. I'm so nervous right now. I'm going to shake you. My whole body is, so please help me out. Today I'm going to sing my traditional song, and I'm going to try Aga. And I'm going to sing the Aga song now. Yes, Thank you all.
Thank you, Sakrinso, for a very beautiful song. Now we will proceed with our program. I request Dr. Vineet sir to invite our next speaker.
Our next speaker is Dr. Gopal Krishna, and presently he is the botanist in Botanical Survey of India headquarters, Kolkata, and he has done his PhD from University of Calcutta, and he also attended training at Natural History Museum, London, United Kingdom, November 2017 to March 2018. His field of specialization is taxonomy of angiosperms and plant nomenclature. His special interest is in family Gesneriaceae plant nomenclature, digitization of herbarium specimens. And he is the fellow of Linnean Society, London, United Kingdom, and a life member of Association of Plant Taxonomy, India, and Society of Ethnobotany, India. And he has described 8 new taxa, 11 proposals submitted to IUCN for amend the code, 2 accepted in Shenzhen code so far. and he has published more than 19 in national, international research papers in peer-reviewed journals. So please proceed, Dr. Gopal Krishna.
Thank you, sir, for brief introduction. I'm audible?
Yes, sir.
Okay, thank you. PPT is visible also?
Yes, sir. It's visible,
Okay,
sir.
thanks. So today I'm going to, good morning to all. So today I'm going to deliver a talk on Herbarium, its history and the methodology. Well, without knowing the history, any topic looks very less interesting. So firstly, we should know what is a herbarium. In fact, when I'm talking about the herbarium, it refers to the two words. One is the collections, what it is housed in a herbarium building, and another referred to the herbarium building. So loosely we are talking that it's a herbarium building, then like a Cal herbarium, some herbarium, then Q herbarium, British Museum herbarium, Harvard University herbarium. So these all are the herbarium building. But when I'm referring the herbarium specimen, that means it's a collection of the dried plant specimen which are stored in a building, and there will be some system of classification. Like in Indian herbaria, you will find that most of the herbaria still we are following the Bentham-Hooker system of classification. But if we talk about the European herbaria or the American herbaria, these all are now following the APG system of classification. Some of the, but still I believe that most of the international herbaria also they are following the APG3 only, because there are certain limitations to transfer into the APG3 to the APG4. So still it is not a well-established system of classification. So most of the European herbaria is still following the APG3 system of classification. If we talk about this herbarium terminology, it was first used by the Tunford in the 1700. The term herbarium is quite interesting. Here now it's although it is a Latin and Greek terminology, but the origin is from the Sanskrit. When people in the ancient time, they were used to feed their cattle from one place to another place. So its meaning, literally meaning was the nourish. So green grass was called as the var, and now it has been modified to the new Latin, it's a herba, and the foria is the old Latin. And this is all about the origin of the herbarium. Now, herbarium, what can be the role of a herbarium? There are lots of number of usage of herbarium. Some of the important, like whenever we are describing a new species, then a herbarium plays a significant role into identity of the species. Then provide material for making a morphological measurement, like when we are writing a description. Then provide local data for the planning field trips, like say for example, if we wanted to visit the Arunachal Pradesh, some of the orchid or the any specialized group of the species. So we should first take the data from the herbarium. In fact, they will give you the past record that where this species is located. If somebody will ask you the Dendrobium imbriatum, where it occurs. So if you are not knowing the herbarium data, it is very difficult to find out which locality it can be. But if you have the already pre-data, so based on that, you can collect in the particular locality. Then serve as a repository of the new collections, serve as a secure repository for the type specimens, then provide material for the DNA analysis. Now, at modern time, what we have seen that old specimens cannot be served a good purpose for the DNA analysis. But for that, better you go for the fresh collection, it will give you the better result. To test the history of usage of the binomials for a given taxon in a given area, provide pollen for the taxonomic, systematic pollination studies as well as the allergic study, provide material for the teaching, like in botany, somebody will give you the project that you go for the flora of the particular areas or the particular fixed location. Then you can go for the herbarium consultation and you can make the data and all that. So then our question comes, why the herbarium, why not if liquid preservation is the useful? The answer is liquid preservation has the several disadvantages. because you also know that in zoological specimens, we are putting the specimens in the formalin solution, and it's very difficult to be, because you are taking out and keeping inside that, and their life will be very less. And then liquid preservation is very expensive method, because you have to purchase the one jar, it's cost around 30-40 rupees, and some chemicals is also very expensive. So putting in this specimens is very difficult. So the dried specimens can be the longer longevity, the more in comparison to the liquid preservatives. So that is, these are some of the reasons we are always used to have the dried specimen in comparison to the liquid preservation, liquid preservative specimens. At present, in the entire world, approximate 3,100 herbaria, recognized herbaria, and the total number of collections, it is estimated as 3,090 million botanical collections are present stored in the herbarium. So here I have used one terminology, it's called the recognized herbaria. To know the recognized herbaria, we should use this website, the Index Herbarium, where you can get access to all that, you can get the data, which herbarium, where it is located. So recognized herbaria means basic differences I'm telling. The herbaria having the acronym, that is called the recognized herbaria, and herbarium don't have any acronym, that is called the non-recognized herbaria. Say for example, Central National Herbarium, the acronym is the CAL. Botanicals of India Arunachal Pradesh Regional Center, acronym is the ARUN. In if we talk about the code, there is the one Article 40.7. This clearly states that for the name of a new species or infrastructure published on or after 1st January 1990, of which type of a specimen or unpublished illustration, the single herbarium collection institute in which type is considered must be specified. That means if you are describing a new species, you must give the correct acronym of a herbarium. Otherwise, this name will be the illegitimate name, not the invalid, it's the illegitimate name. Then another is called the non-recognized herbarium, means there will be no acronym. Say for example, Institute of Bioresources and Sustainable Development Herbarium in Manipur, this herbarium is not recognized. Then now question comes, if it is a non-recognized, how to cite that recognized non-recognized herbarium? So answer is Institute of Bioresources and Sustainable Development Herbarium. If we will talk, then we have to add a last term herbarium. That means now it is clearly stating that the Institute of Bioresources and Sustainable Development is the organizations or the institute, and herbarium, whatever the specimens is stored there, they all are the belonging to that. But if somebody use that IBSD, because it's means popularly the people are calling it as the IBSD, then it is wrong, because if we see that index herbarium, then you will find that IBSD stands for the this Chinese herbarium. So that means you are referring a wrong herbarium, and people may not find the specimen. So this is the advantage and disadvantage of the recognized and unrecognized herbarium. If we talk about the historical history of this herbarium, long back in the 16th century, Lusagini was a professor in the Italy, Padua. So he started this technique, and in the garden, because he was used to collect all the medicinal plants and putting on the book-like structures. Means one side he was writing a description, and another side he was putting the specimens. So it was referring to that. You may say that in the practical record, if you can remember, one side, left side, we are drawing and right side we are giving the comments on that. So it was similar way they were preparing that kind of herbarium, but no longer existence of these specimens. Then this is the place, Italy, Padua, where he has started this. Later, there is two very famous students. One is called Andrea Sigelpino, and another is Alvandro. So they also carried out the same process. In you can see that how the specimens were used. Here, this specimen, a little write-up is here. Here you can see that the specimen is here, some writing is here. Here there again a specimen is here, and these are some of the descriptions are here. So this is the old style, and until this is another example. But in 17th century, when Carolus Linnaeus came, he started some different practice. So what he did, he put the specimen on a single sheet. These all of the Linnaean Society collections. You can see the single sheet, the smoother collections, and From then people find that this is a more convenient, and people can use, a researcher can use the specimens very easily. And now what practice we are doing, in fact, this practice has been started by the lineage. If we talk about the initiation of herbarium preparation in India, it was started in the by the William Roxburgh, and this is the first our herbarium building in India, still existing in our Indian Botanic AGC Bose Indian Botanic Garden. It is built in the 1793, and here he was used to keep the specimens. But looking at the climate of the West Bengal, the specimen, keeping the specimens was very difficult. So in replacement of that, he was used to make the live form of the drawings with what's called the Roxburgh icon. So all the specimen has been sent back to the European herbaria of Roxburgh collection. Exceptionally, except some 70 collections have been remain in our Calcutta herbarium. So still we have the 70 number of collections of the William Roxburgh. Later on in 1890, another building was prepared by the George King, and this is called our second herbarium building in India. On the first floor, they were used to keep the specimens, and on the second floor, it was used to keep the library. And this is the modern herbaria, means it was built in the 1971-72, and here you can see this is the central herbarium, and these are some cabinets, and the wooden cabinets, steel cabinets. Then now we coming to the herbarium methodology, means how we are making this herbarium. It's not a easy task. That means you go to the field and your herbarium will be prepared. We need the continuous effort. Say for example, you may see there in the 5-6 pictures. Here we went for the collections. Then we are putting into the blotting sheet or the newspaper. And after some treatment, the third day you can see the collections are like this. And finally, this is the ready for the mountings. So practically how this is possible. So in field, we are went for the collection and field photography. This is very important nowadays. Then processing and mounting, dissection in the field, and lastly, identification of the specimens. So these are some of the equipments which is very important while we are going to the field trip. This GPS, nowadays mobile, if you have the internet access during the field, then it can be the replacement of the GPS, but the error will be more in comparison to the GPS, what the Garmin GPS have in the facility. So these are some of the collection pouch, why we are keeping in the field. Say for example, you are collecting in a field 50 collections. So what I am doing, so A4 size of, or more bigger than the A4 size of zipper, whatever collections we are collecting, we are putting in one zipper. So one collection, one zipper, one collection, one zipper, and this is the bigger size of the plastic bag or carry collection bag, where we are putting all the specimen inside that. So if it is a very fast process in comparison to the keeping the tagging in the field, because it takes time. So what you can do, collect the specimen in the field and put in this zipper pouch and put the one tag inside in the field itself. So this will save your time, and this is the field book. You can here fancy, and these are some of the collection for the pickle, making the pickle of the specimens, because some of the specimens are in the very delicate flowers and quickly it dried, and once it is stick together, it's very difficult to take it out. And better to make a pickle like a pickle of the specimen, and later you can extend. Already I mentioned it, blotting sheet, polythene bags, and the specimen tubes. So what are our objectives of the collection of the specimens? The botanical collections are made up for the building a new herbaria, for the enriching old herbaria. Then collection plant materials for writing a flora, collecting live materials for introduction in the garden or habitations, collecting materials for particular genus species for the research work, for the analysis, then collecting large quantity for the trade, ethnobotanical studies. So these are some of the important objectives for the collection of the herbarium specimens. Now question comes, how many numbers we should collect? In fact, a person who is very expert in their field, like those who are expert in their field, so they have an exact idea whether the species is threatened or not. If it is threatened, better to avoid the collections if already it is existing in the herbarium, or if it is not existing in your herbarium. So you can make a one herbarium collection, but don't say for example, if it is a herbaceous, don't uproot from the, means don't dig out from the root. So better you take a twig and make the herbarium specimens. If it is a tree species, there is no problem. You can make 3-4 twig, there is no problem. But in case of the herbaceous collections, you need to be very careful while we are collecting. If say for example, if the specimen is a lantana, we need not to go for the 5-6 collection. A single collection is sufficient unless it is not different from the existing collections. So this all depends upon the whether we are beginner or if we are very experts in the particular group. So if we are a beginner, in that case, if you can see in the particular area, the collections are very less. So try to avoid the multiple collections. To go for the single or double collection, this is the sufficient for that. If it is abundant in your area, then you can go for the multiple collection. There is no problem there. But 3 is the sufficient for the database. If you can remember, in the past, this is the Second World War image. What happened? This image is of the British Museum. In the Second World War, in aggression of that, Germany bombarded on the herbarium of the top of the herbarium, means herbarium building. And they wanted to, in fact, destroy the entire herbarium. Somehow, only top storage buildings have been destroyed, and the many of the grasses specimen has been damaged. This is just for the information. Then photography is very important. See that in the field we don't have any scale. So what I did, I used this 15 big point to make the photo plate and all. And this is the Galeola. So whenever we have in the such kind of collections, if it is interesting, because I was not aware of this genus earlier, then we have made for the photo. We went for the photo plate, and in the field itself we have dissected all these things and we have prepared a photo plate. So likewise, if you are not aware of a species, whether this is a what is species it is in actual, then you can also go for the making the photo plate in the field itself. It's a better idea rather than you are we bring the specimens in your camp and you will go for the photo plate, because live collection has the better option for the making photo plates. Astrobalanthes, in the case of astrobalanthes, it's very important because once the species you put in the plotting sheet or the newspaper, the corolla tube stick together, and it's very difficult to go for the dissection. So in particular case like impatiens, astrobalanthes, some of the orchidaceae members, you will find very difficult to study after sticking the petals and all. So better you do it everything in the field itself. Then field notes, it's very important because that nowadays we having the access of the mobile and be a good camera, but in past the people don't have such kind of things. So what they were using, they were whatever data they were seeing in the field they are noting down. And say for example, flower colors. If you see the old herbarium, very few herbarium specimens you will find that what color of the petal was there, because they have not recorded at that time, and later on they forgot all these things, and you will not find such kind of. But if you will go for the field note, then you will find that okay, they have mentioned about that the flower was the yellow, white, blue, whatever things. So some of the things you cannot recall, but in the field itself, but you can write in the field as a field note. Already I mentioned about this tagging, how we are doing in the field. So this, after coming to the camp, now our job is to preservation of the specimens. It's very important. So basically 2 methods we are applying here in India. One is called the dry preservation method, another is called the wet preservation method. Wet preservation. So wherever there is a rainfall is more, like Northeast India, Western Ghat, there is the rainfall is more. So these area you try to follow the wet preservation method. And if the state like Bihar, Jharkhand, then Madhya Pradesh, Uttar Pradesh, where the rail, Gujarat, this area having the very less rainfall, you can go for the dry preservation method. What is in that? Basically, what we are doing, alcohol 90% and formaldehyde we are adding the 10%. This solution and putting in the tray. After putting in this tray, we are dipping the live collections and putting into the blotters. And after second day, after doing this, you will find that the species, this specimen is drying very rapidly. The reason behind that, that the leaf surface having the stomata. So after putting inside the alcohol enter into the mesophyll tissue and the water content dehydrated, because you know that when alcohol combines with the water, they become the vapor. So the specimen which will be dry in the 5 days or 6 days, it will be dry in the 2 days. So this is the advantage of the wet preservation method, and it is also applicable in that case where the abscission layer is rapidly formed, like family Euphorbiaceae, Leguminaceae. So these collections you will not find the leaves are intact with the specimen. So in that case also this is very useful. In the wet preservation method, simply we are putting the specimens into the blotting sheet or the newspaper, and after 5-6 days it becomes dry. But very important thing, when we are putting the specimen inside the blotters or the newspaper, you should go for the change the blotting sheet and the newspaper every day. This will keep your specimens beautiful, and drying of the specimens will be rapid. In comparison to the specimens are put into the blotting sheet for the 5 days, 6 days, we are not changing. Then the chances of the decay of the damaging of the specimens are very more in comparison to the species which are drying. So this is all the things. Yeah. Day after the alcohol and formaldehyde treatment, you can see the changes. Just I am talking about the 1 day later, you can see the changes in the specimens. Live collections now it turn into the little yellowish color, and most of the water has been gone. So after that, we are putting into the herbarium press. That's called the hot pressing. And after coming to the herbarium, you have the access of this ball press. It is a heavy press. After drying of the specimen, then we are dipping the inside again the second time alcohol treatment. So what is the benefit of alcohol treatment? The whatever the germs will be there, means insects or bug will be there, all will be damaged with the alcohol treatment. So in past the people were using the mercury chloride, but at present it is the banned in the most of the herbaria. In particularly the European herbaria, they never used to of the mercury chloride treatment. The reason behind it is not essential in that particular area, because there is the rainfall is the less in comparison to the tropical country, and the weather is the first factor where they were not using any mercury chloride treatment. But here in tropical country, where you will find the difficulties in that. So to maintain the insect free, we are using the mercury chloride. But still, I think BSI, Calcutta Herbarium, they are not, scale Herbarium, they are not using any mercury chloride. What are the replacement of that? So better to put the specimens dry, means humidity free. This will be make your herbarium more longevity. So there is another options using of the silica gel. We have to put the silica gel inside the herbarium cabinet, and you know that once they will retain they will capture all the humidity, their color will be changed and white to bluish color. Again, you put the silica gel into the into sunlight, and the color will be again changed. Once the humidity will go off, it will be turned into the white. That means this is the identity of the silica gel, whether they have retained the humidity or not. So this technique is very effective in the most of the tropical herbaria. In the bryophyte, there is no need to go for any alcohol treatment or any other chemical treatment. The bryophyte plant have their own capacity to protect themselves, and in that case, we are putting into the small zipper means this pouch or capsule. And some minute plants like Wolffia, Lemna, these species are very minute and microscopic. So there is some different treatment. Here you can go for the ethyl alcohol, glacial acetic acid, and formaldehyde 40%, and water. These three compositions are very effective in the case of the minute plants. The collection of pteridophytes, you know that the collections is very tricky. I am not expert on the pteridophyte. Vinit sir is an expert on that group, and he may guide you that how to collect the ferns, whether the root is important or not, means the rhizome is important or not. So it depends upon, but when we are going for the mounting, then we should be very precautious, because the lower surface having the sporangium. So sporangium is having the very big role in the identification. So on that point of view, I can say that in mounting, you have to be very precautious. So both surfaces should be visible. In a big collection like bamboo, then palm, these are the huge collections. So how to collect of that plant? It is you cannot make a one leaf herbarium specimen because of the size. In that case, what we are doing, you make the pieces of a leaf. Say for example, a leaf segment having into the 20 pieces. So all 20 collections will be considered as the one collections, but you have to mention clearly like 1 by 1, so 1 by 20, 2 by 20, 3 by 20. That means people may understand that this particular herbarium collection having the 20 sets and all together will be called as a single herbarium collections. So in that case, again, there is the alternate options. You take the photograph, good photograph of that, and you make a small piece of a herbarium specimen, paste on that, and right side you can paste the photograph. So from the photograph with the scale, so people may understand that, okay, this plant will be looks like this one, their size will be this one, their shape will be this one, and this is the measurement of this particular things, and the sample small piece will be the representative. So two way you have, either you take the paste the photograph and along with the this piece of the herbarium collection. These are the bamboo collection. You know that in the case of the bamboo, the culm seed having the very important characters, and it helps in the play a significant role in the identification of that. So in bamboo, don't collect only leaf and the inflorescence because bamboo flowers very rarely, most of the species, and if you don't have the flower, the characters are very less. So to make all these things, they are critically they are going through the culm seed and the culm of a bamboo, so that they can determine what species it is. So I was talking about this. This is the pandanus species. So if we are making a herbarium of a pandanus, it's very difficult. So you cut a small piece of this particular pandanus and paste on the herbarium, and you take a good photographs and you paste over there. So this is the sufficient. For the bulky specimens, why? What is the important in that? If you have the bulky specimen, like the eagle marvelous fruits are the very bigger inside. Basically, the concept is that if the thickness is the more than 1.5 centimeter, so in that case, better to make a carpological box. So what is the carpological means? This fruit box. It may be the depend upon the what size of the fruit you have. So manually you can prepare that herbarium boxes. So here, what you have to do, put the fruits inside this box and level, what level is the representing here, same level will be here. And here you have to put under notes that for the fruit, you have to see the carpological collections. I have not seen these collections very good number we have at the Calcutta Herbarium. But in Q Herbaria, I have seen that they have their separate carpological collections, not in Indian Herbarium I'm talking. Already I mentioned that don't use the this very health hazardous chemicals like mercury chloride. So better go for the alcohol treatment as many as times. It will be very effective and useful. Then mounting of the specimen. For the mounting, we need these are items, very important. And standard size of the mounting boards will be that 42 cross 28 centimeter in India, but in European herbaria, they have the 42 cross 29, somewhere it is the 42 cross 26, 44 cross 29 centimeter. But better we have to follow the 42 cross 28 centimeter. This is the standard size. On the rightmost side, there will be a herbarium label. It is of the 11 centimeter cross the 10 centimeter. In fact, why it is on the right side, the reason behind, because if you will see the when we are opening a cabinet, from there itself we can take the help and we can see that whether this is specimens, whether it is belonging to the same species what we are searching or not. So for the user-friendly purpose, we are using the right side herbarium label. And this is a typical herbarium label what we are using at the Central National Herbarium, where we are flora of collection number, date, family. So this depend upon your need. So if somebody is working on the pollen, they may have some pollen data also, they can not over here. It is depend upon their usage. In the adhesive, there are the 5-6 substitutes we have. In past, we were using the here animal glue. In fact, in animal glue, what we are doing, the Swiss and plus the animal hoops were using. So now it is a completely banned in India. And now what we are using, in place of that, we are using the Fevicol. In fact, other than the Fevicol, we have some options like polyvinyl acetate, methyl cellulose, sodium carbomethyl cellulose. And one glue is called the Crucial gel. It's quite expensive. It is the animal cellulosic glue, and it's very used for the archival materials. In fact, what are the reason behind that? In Fevicol, once you are pasted the herbarium specimen, you cannot take out very easily. But in case of the Crucial gel, it is a reversible. Once you are putting the water on that surface, it can be taken out easily. So for the remounting purpose, I think the best method is go for the methyl cellulose, but it's quite expensive and the archival materials. But normal purpose, you can use the Fevicol, what entire world is now following. And wherever you will find the surface stitching is very important. You can see that here, if stem is more than 1 centimeter thick, you have to go for the stitching, because the Fevicol on the lower surface, it cannot hold for the longer period. After a time, what you will find, the surface has been coming out. So it's very difficult. So better to go for stitching of the specimen. It is still in the trend, and we have to apply that technique. So after that, we have to go for the identification of the specimens, and after the identification, we should incorporate the specimen in the proper way. So in incorporation, we have to come to know about, we should know about what is the genus cover, then species cover. These are some of the standard protocol what we are following at the BSI herbarium. This is our type herbarium. So now, if we talk about the arrangement of the herbarium at Calcutta herbarium or the BSI herbarium, most of the BSI herbarium, we are following this Panther system of classification with slight modifications. Here, these are the techniques what we are for the BSI herbarium. Calcutta CNS herbarium is arranged. Firstly, we will have the Indian continent region, this India, Pakistan, Nepal, Bhutan, Bangladesh, Sri Lanka, then this Eastern India and India into the South America. So likewise, we having the 12 geographical, 5 geographical region collection we having at the Central National Herbarium. So this is a cabinet. This is the still cabinet, you can say that. And here in each cabinet, you will find the 60 pigeonholes are there in the case of the steel almirah, and in case of the wooden almirah, there is the 130. This is the example of the wooden cabinets. So if you see the genus cover, you will find the first name is the genus, followed by the author of the genus. Then GP means Genda plantarum. The genus number 22 belongs to the Genda plantarum. And this geographical region 1 by 1 represents to the India. And another already has said that some of the herbaria in Europe, they are following the APT system of classification. Like this is the British Museum, this is the Kew Herbarium. Thank you very much. So this is all about the herbarium methodology and history.
Okay, thank you, Gopal sir. We can take few questions from our audience.
Yeah, please.
Anyone? Yes, there is one from Himalayan University.
Good afternoon, sir. My name is Kaling Zama. My question is how to prepare herbarium specimen for SEM, scanning electron microscopy.
Electron microscopic means herbarium. I don't know about this one. Whether there's any why you want to a different kind of herbarium preparation for the SEM?
Yes, sir.
I mean, what portion you are going for the scanning?
Of course, sir, you have already said that conservation of specimen like
dry conservations, wet conservations. But I'm going to ask about the SEM, how to prepare specimen for SEM.
Well, okay, I got you. In fact, you are asking that if you wanted to the same study,
yes, sir.
So what kind of preservation method we should apply? This is the question?
Yes, sir.
Okay. I think, the pollen, no?
Yes.
Pollen will not change anything. Even the chemical, if you are using, the pollen will not change. So the older herbarium specimens also serve the purpose for the pollen study. I think Aarti ma'am is the right person to answer this one. Ma'am, can you please answer this one?
Yes, if you have, with the permission of the speaker, I would like to elaborate on this. See, for SEM, our SEM work is always targeted. Whether we want to take the SEM of a leaf surface, or we want the pollen grains, or we want the seeds SEM. So these are the basic structures for which SEM is usually utilized. If we go for pollen SEM studies, there is a set of procedure. We extract one anther from the sheet, put it in alcohol, and then it is passed through an acetylysis method. Now this acetylysis method is elaborated. It has got 10 steps of procedures, after which the protoplasm from the pollen grain passes out through the apertures, and the pollen ball becomes like an empty ball. And then we put it in alcohol to finally dehydrate it in 100% alcohol and mount it on stubs. So this is what I'm telling you about the pollen. It is mounted on different photographs are taken. The ball remains intact. Internal protoplasm flows out. So if we get a very clear picture. Then if you want to go for the seed, there is no need to empty the seed from itself on the seed surface. So as it is, the seed can be extracted from the sheet, placed on the stub directly, passed through a series of alcohol, 30%, 70%, photographed without any following any intricate procedure. Then if we want to go for the leaf sample, the leaf surface has again is involving some methods like direct peeling can be done, or with nail polish we are sometimes peeling away the surface, and the peeled surface properly on slide with cover slip, and with the cover slip in cavity blocks it has to pass through series of alcohol and very gentle. So for on specimen which has to be photographed. So that's it.
Thank you, ma'am.
Yeah.
Ma'am, actually his question was how to make a herbarium specimen. In fact, herbarium cannot.
No, herbarium specimen. Yeah, okay. If he's asking for herbarium specimen, what specimen we are making itself?
Yes.
From where procedure has to be followed for SEM. There is no separate herbarium procedure for the plant part.
Yes. Thanks, ma'am.
Right. Every plant part is preserved in the form of herbarium. You can use it.
Okay, so you got the answer.
Am I clear to the? Yeah. Anything else on this point?
Any other question?
I guess that there is no questions from the audience. So once again, thank you, Gopal sir and Aarti ma'am for clarifying the questions. Anyway,
Okay.
we will take a.
Yeah. Then, with permission of Vinit sir, I'm leaving because I'm busy with I'm some at university. So
okay, Doctor. Okay, no problem.
Okay.
Okay, thank you. Thanks for sharing your valuable time for us.
Okay.
And
Thanks,
I hope online, offline students they have taken interest and
yes.
they have
in
learned.
In case of anybody has question regarding the herbarium methodology and all, I can share this PPT to the Vinit sir and you can collect from them. Okay. So I hope this may be the useful to you, and you are always welcome because we have the very limited time. I haven't talked on the what kind of papers should be there, what kind of threads should be there. So these are some different aspects of the herbarium preparation. Simply in the brief, because the limitation of the time, I could not elaborate on that part. So if you have any query regarding what kind of paper, what kind of things you should use for the making a better herbarium, not for the general herbarium, because at the university level, people are using this some ordinary kind of paper. It is not good for the preservation of a specimen. Okay, thank you very much.
It was very informative lecture, Dr. Gopalji. Thank you.
So we will take a short tea break for 10 minutes here, and we will resume the program at 12:15. 12:15, sir? 12:15. 12:15, okay. So I request everyone to join the program at 12:15 p.m.
கொண்டு வா இங்க தம்பி இங்க ஒரு பால் பால் Mumbai. अय्यो. ते काय मेसेज का तेवढं ही लाडकी कुडाला राहता अय्यो.
So we will start after 3 minutes.
तो फिर से आप सबका स्वागत है हमारा ट्री ब्रेक के बाद। तो हमारा जो नेक्स्ट टॉपिक होगा वो है एम्पावरिंग प्लांट टेक्सोनॉमी यूजिंग मॉलिक्यूलर टूल्स। तो हमारे जो सर है उसके बारे में हम और डिटेल में आपको डिटेल में जो है हमारे विनीत सर आपको बताएंगे। सो आई वुड लाइक टू वेलकम आवर विनीत सर।
नमस्कार आप सभी को, गुड आफ्टरनून। तो नाउ नेक्स्ट स्पीकर इज डॉक्टर रितेश चौधरी। मैं आप सभी को बताना चाहूंगा कि रितेश सर ने यहाँ BSI APRC में वो रिसर्च स्कॉलर थे और यहाँ पर उन्होंने रिसर्च किया। तो नाउ he is the scientist in Agarkar Research Institute. Now I'm giving the brief introduction of Dr. Ritesh Kumar Chaudhary. He is presently working as scientist at Agarkar Research Institute, Pune. And after finishing his PhD from the Botanical Survey of India in Rajiv Gandhi University, Itanagar, he worked as a research associate at GKVK Bengaluru for 1 year. In 2010, he received the prestigious Korea Science and Engineering Foundation postdoctorate fellowship. He moved to Korea Research Institute of Bioscience and Biotechnology, Daejeon, South Korea, for pursuing his doctoral research. In 2013, he returned to India and joined Agarkar Research Institute as a scientist, having experience of more than 18 years as a plant taxonomist. He has worked in all 4 biodiversity hotspots in India. While working at BSI Itanagar, he has completed the flora of one of the most arduous region, Upper Siang district of Arunachal Pradesh. Apart from this, he has also worked on the flora of Honban Nature Reserve in Vietnam. He has described 13 new plant species and reported 21 new records from India and Vietnam. He is one of the few taxonomists of India who is currently studying the molecular phylogeny of Indian plants. He has more than 85 research publications in Journal of International Repute and 3 books to his research credit. Some of the key publications in taxonomy are the genus Caperes in India and floristic diversity of Honba Nature Reserve in Vietnam. He serves on the board of studies of SP Pune University's Botany Department. He is an adjunct faculty at SP Pune University, Pune, where he teaches molecular taxonomy to the MSc students. He is also serving as a research advisory committee member for Nehruji Godrej Center for Plant Research, Sriwal, Maharashtra, India. He also works as a reviewer of many reputed national and international journals and an editor of the Journal of Economic and Taxonomic Botany and Journal of Biology. He has been awarded Professor B.V. Sivarajan Gold Medal in the year 2020 and the Best International Researcher KRIBBS South Korea in 2011 for his contributions to plant taxonomy. Thanks, Dr. Ritesh Chaudhary ji. I invite you to proceed a lecture.
Thank you so much, Dr. Vineet. And I would like to share my PPT first, and then I will. Are you able to see my screen? My screen is visible to you. Could you please confirm?
It is visible.
Okay.
Yes.
Thank you so much. So, thank you so much for the introduction, kind words. Dr. Vineet sir, I'm calling him sir because he is my senior while I was working in BSI, and I know him for quite a long time. And at that time, he had made several visits to Arunachal Pradesh, and I have a special, the Arunachal Pradesh actually have a special place in my heart because I have done my PhD from that place. I have done PhD on the flora of Upper Siang district. So, but anyway, right now I'm doing something else, not the floristic work, but still associated with plant taxonomy. During my floristic works, I realized that with the changing time, we need to upgrade this plant taxonomy skills. And with the help of molecular tools or the advanced tools and techniques, we can empower it more and more. So today's topic of the talk is the same, empowering plant taxonomy using molecular tools. And before I go ahead, I would like to introduce a little bit about my research institute where I'm presently working. I work at Agharkar Research Institute, which is based in Pune. And it's an autonomous institute under Department of Science and Technology, Government of India. And here we actually work under 6 different themes. We have 6 different groups. One group where I'm working is biodiversity and paleobiology group, one of the biggest groups here. Another group is there, which is known as bioprospecting group. We have bioenergy group. We have nano biosciences group. We have developmental biology group also, where we work under 3 different model organisms, zebrafish, Drosophila, and Hydra. And we have another big group on genetics and plant breeding, where we work on 3 different crops, grapes, wheat, and soybean. And we also have several PhD vacancies. In case any of you interested in coming here and want to do PhD at Agharkar Research Institute, please keep an eye on the website, which I have given here, www.aripune.org. And you can just keep looking at the website, and we have several openings. Day to day, we have several openings, so you can just keep an eye for the updates. And in my group, actually, it's not very kind of hardcore taxonomic studies we are doing here. Rather, we are doing kind of multitasking here. We do document flora and fauna. And then we also make effort to understand evolution. We also work towards exploring biodiversity for human welfare. We are also working towards conservation of the plants. And also whatever we are doing, we are trying to communicate that. And we are also developing human resources. And we have capacity building programs as well. So as a taxonomist, I would like to say that we have been describing species, new species of plants and animals for quite a long time. But you will be surprised to know that the species discovery pattern has increased much more than before. In the whole 250 years of its history, it has increased a lot. Now we are identifying and describing new species at a much faster rate. And during the last year itself, more than 18,000 new species of plants and animals were described. And approximately 25% of all known amphibians were discovered over the last 10 years. And also within the last 2 decades, the number of known mammals have also increased to more than 10%. And you might be asking, like, why it is so? So many of the discoveries actually resulted because we have many new tools and techniques available with us. And the recent tools and techniques, like you can do a lot of things, which I'm going to describe today. So as we know, this plant taxonomy has seen several phases, which I think my earlier speakers have already been describing it. The first phase, which was kind of exploratory phase, where we used to discover, describe, name, and identify the plants and classify them, was called an exploratory or pioneer phase. Later on, we shifted to consolidation or systematic phase, where we are just started doing that, writing floras, monograph, and started forming a classification system. Further, we moved with course of time, and we entered into experimental or biosystematic phase, where we started understanding evolution using chemical data, numerical data, cytological data, and what's not. But presently, the era in which we are is called an encyclopedic or holotaxonomic phase. I'm referring to this Davis paper, which was published long back in 1963. And here, what we are doing, we are following a holo approach. Like, we are trying to classify animals or plants taking data from multiple sources. It's not like restricted to just morphology. So if you see these 2 phases, the earlier phases, we used to name as alpha taxonomy. And then what we are doing presently is kind of, we are doing omega taxonomy. This is called, often referred as omega taxonomy. But as something new comes, apprehensions also arises, right? So if in the last 2 decades, so many papers started coming, so many research works started coming, where they used a lot of new data from molecular science. So many papers also got published, and like the paper, you can see that people have so many kind of apprehension, whether we are molecularizing the taxonomy, whether what are the concerns of molecular taxonomy. So there were several concerns. So what would be the promise of this DNA taxonomy, they named also. But before we think so, we have to see a paper which was published by Hortel et al. In 2015. The paper entitled 7 Shortfalls That Beset Large-Scale Knowledge on Biodiversity. So here they have mentioned, unless, they have mentioned about 7 different shortfalls. Called Linnean shortfall, Wallacean shortfall, Prestonian. So what is this? So they are saying unless you describe each and every species on Earth, there's a lacuna, there is a shortfall in your study. Unless you know about their distribution patterns, there is a shortfall. Unless you know about the ecological interaction, there is a shortfall. Unless you know about their habitat, evolution, and so many other things, you can just go through this paper. There is a shortfall in your biodiversity study. So here comes molecular tools or molecular taxonomy, which we call. So if we take help of this molecular data, definitely there are so many shortfalls we'll be able to overcome. Right? And I think during the last 2 decades, there were also a kind of branch of taxonomy was proposed called numerical taxonomy, where what they used to do, they are taking help of the quantitative data. They will measure the stamen length and length of the flower, length of the stem, and so many other features, and they will prepare a phenogram. And based on those characters, this used to measure it. And based on that, they used to develop a phenogram based on morphological similarity. It will show you a tree and it will try to tell you that what are the relationships between A and C, what is the relationship between A and B or a D. But this had a major issue. You can see the first tree which you can see was prepared using the quantitative data or numerical taxonomy. Here you can see this particular tree is putting Opuntia and Euphorbia together. We all know that Opuntia belongs to Cactaceae and Euphorbia belongs to Euphorbiaceae, 2 different families. The same way, this belongs to Euphorbiaceae, Pereschia belongs to Cactaceae. But looking at the morphological similarity or the quantitative data, the tree phenogram generated using numerical taxonomy, it is putting them together, which is not the correct one, right? Opuntia and Euphorbia cannot be so close together like Pereschia and Codium they put. So if you are using molecular data, what we do here, we are using DNA of those plants and try to compare the DNA. You can see how correctly it has been placed. And also sometimes what happens, there is an enigma of convergent evolution. You can see these 2 plants, you can see one belongs to Euphorbiaceae and this one belongs to Cactaceae. And because of morphological similarity, and I just wanted to tell about this convergent evolution. What happens because of the same ecological pressure, some of the plants, they start developing similar feature to the plants which are growing in the similar habitat. So here, the particular Euphorbia, which is not cactus, and because it is growing in desert environment, it has also grown up certain characters which are very similar to cactus. And because of this, and these characters are very deceptive. You will not be able to, at one go, you will not be able to find whether this is a Cactaceae, whether this is a Euphorbiaceae, right? So such kind of problems can be resolved using molecular data. Then also there is an enigma of cryptic species. You can see like one plant in the field if you go, and one plant if you see just if you just isolate DNA from multiple individuals of a single species, what happens, DNA cryptic species we call to those species actually which are morphologically same, but at molecular level they are different. So there are many papers have come up right now, and this problem has never been addressed by morphological taxonomy. So using molecular tools, you can address this issue as well. So what we do here in molecular systematics, or what kind of molecular tools we use in plant taxonomy, here we are making use of plant genomes. Where we are going to find them? Genomes we are going to find in 3 places in plant. One is either nucleus, or in chloroplast, or mitochondria, right? And we are taking isolating DNA from that and try to understand the composition of those genomes. And in the last 10 years also, we have so many markers available. And there are three different kind of markers available. One is called morphology-based marker, another is biochemical marker, and third one is our molecular markers. Today we are going to focus on these molecular markers only. Biochemical markers are isozymes, and morphological, just appearance of, with the help of appearance, we can select certain seeds, that is called morphological marker. But today we are going to focus more on molecular markers, which are, for convenience, we have divided them in two types. One is hybridization based, another is called PCR based. One of the example of hybridization based marker is RFLP, whereas in PCR based, there are several markers available, RATD, AFLP, SSR, and so many others are available. But among the PCR-based markers, I would call this as a superhero of molecular markers called site-specific markers. And one of the most popular among them is ITS, which is a kind of transcribed spacer region, and the full form of this internal transcribed spacer. And it is nothing but a small region in the nucleus, and almost approximately 600 to 800 base pairs long, and they are very much species specific. If you are just trying to isolate this particular region, if you're isolating this particular IT, you can see the placement here. If you just isolate this particular region and try to read the sequences, you will find for two species, the sequence will be two different ones, not the same. Same with chloroplast also, we have several regions called RBCL, PSBA, PSBD, ATPA. So many regions are there which are very species specific, where they're very conserved for those species. They're very small in size, and nowadays with the help of next generation sequencing, it has become possible that you can sequence the whole chloroplast genome also with a very little amount of money. Then we have certain regions in mitochondria as well, which is approximately 200 to 2500 kbp long. And then there are several regions called tRNA, F, and so many others like ATP1, ATPA, MAT-R, MAT-K. These regions are there. If you just isolate these particular small regions from mitochondria and just try to read their sequences, they'll be different for different plants. They'll not be the same. Then, as you can see, in the last 2 decades, there have been several papers published. First one was published in PNS. It was long back, old paper, but how these particular markers have helped us in empowering this plant taxonomy. With the help of these markers, we have resolved so many species complexes. We can understand the phylogeny, we can understand the evolution, and so many other things. Even they can be used as a barcode for that particular plant. I would give you an example of this particular plant group called Eriocolon, on which we have worked, and this is Ashwini, Dr. Ashwini, and she has finished her PhD in my lab. And this is a plant group called Eriocolon, very common in the Western Ghats, not in the Eastern Himalaya. Eastern Himalaya is also there, but compared to Eastern Himalaya, they are kind of 60-40 ratio. They are distributed. We have almost more than 100 species in India. Out of that, 60 species are present in Western Ghats, approximately 40 in the Eastern Himalayas. On the herbarium sheet, if you see them, they look alike. Can you identify them? Like, they look so small here. And if you want to identify them, one of the best thing about this, many of the species have been proved of a lot of medicinal use. So these figures, these heads are from 3 different species, not the same species. And one species has been proved good, effective as a neuroprotectant. And many of them have shown significant therapeutic against, like one species has also been found as an anti-cancerous value. So if you want to identify this species, it's hell a lot of job. So if you want to identify this, first you have to dissect those tiny head, and then on that head, you have to dissect out this involucral bract first. That is not enough. Then you have to dissect out floral bracts, which are, you can see the size, and then male flower, not enough, and then try to understand the structure of the morphological structure of the female flower. And then not only that, you have to check the structure of the seeds, which are very minute, you can see there. So what you're going to see do there, you are going to check at a light microscope. That is also not enough. You have to go for SEM, and that is also not enough. And in those seed, you have to find the structure of the appendages on the seed. So once you check the structure of the appendages, you can see the variations in the appendages of the seeds. Once you see the appendages, then only you can confirm the identity of an Eriocolon species. So that's why, because of this, and also not only that, in these species or these particular Eriocolon is so complicated genus that they show high intraspecific variability. Means within a same species you'll get a lot of variation, and between two species very few variations are there. Because of this, they're always referred as a taxonomist nightmare. And because of these all problems, Dr. Hooker, J.D. Hooker, when he was writing Flora of British India, he had mentioned that they are the most difficult of classification, presenting no good sectional characters. So we thought of taking up this challenge using molecular data. So we made a lot of survey in the whole Western Ghats. We started it from our northern Western Ghat with the help of our institutional funding. And after that, we got more support from the institute, and we extended our work from till Eastern Himalaya also. We made a lot of collections from Meghalaya and also some other regions of Northeast India. So from Western Ghat, we collected more than 550 accessions. Just for confirmation, we collected multiple accessions, and finally this work was published in a good journal called Systematics and Biodiversity. You can find this paper. If you don't want to read it, you can just send me a request on ResearchGate or through mail. I will send you the paper, where what we have tried to understand, the evolution of or phylogeny of this particular group called Eriocolon, how one species is related with other. Not only this, with the help of molecular data, and this was based on two different markers. One is called ITS, which is a nuclear marker, another called TRNLF, which is a chloroplast region. And this, because based on this, we could synonymize almost 7 different species, because they were kind of, you can say, pseudo species. They were because of some sort of morphological differences, people have published as a distinct species. But with the help of molecular tools, we found that they are not good species. They are actually those species were earlier already described. Not only this, we could reinstate one species called Eriocolon rhodi with the help of molecular data. And then we found there's a little congruence between the morphology-based classification and this molecular-based classification was a little different. I would tell about another story of a Caparis group. We got some funding support from SCRB DST for carrying out this work. This particular plant group you might have seen in Arunachal also. There are so many species there, and this is called caper also in English, and care we call in Hindi. And my student, Mr. Satish Maurya, he just got doctorate on this degree. So we perform phylogenetic studies of these particular plant group. Why they're so important? You might have seen this Vedic medicine called Leaf 52. If you see the label, one of the major component of this particular medicine is this Caparis spinosa. Almost you can see 130 milligram is of Caparis spinosa they have used in Leaf 52. And then the other species also medicinally very important. Not only this, the fruit of this particular group of many of the species, they are eaten as a pickle. Though they are not very popular in India, they are very popular in Europe. And also it's slowly getting popular in India as well. In Maharashtra, many people, they eat the fruit as well. So we decided not only to do this taxonomic revision using this molecular tools, but we try to understand how those morphological characters have evolved in due course of time. And then also we wanted to understand the phylogeographic pattern, how this particular Caparis group have evolved in different continents. So with the help of molecular data, we could find that. And there was a hypothesis called Jacobs hypothesis, where the famous taxonomist Jacobs, he had proposed that the particular genus, some of the species, they originated in the peninsular India part. So we just wanted to understand that, whether with the help of molecular data, we'll be able to understand that or not. And also we wanted to develop DNA barcodes for many of the species. Why that was so important? Because we have seen that they are medicinally very important. But what happens in Ayurvedic medicines, there are a lot of adulteration have been reported. So if you want to consume one particular species for some particular ailment, what happens? They will give you some different species that will not work out for that one. That's why identity of that species is very important. And that becomes crucial when you're simply given a leaf and ask, okay, can you identify this without having flower? So with the help of morphotaxonomy, that is not possible. If I simply give you a leaf asking identification of a particular plant, can you do that? No. But if you have a DNA barcode available for that, even if you have very small amount of leaf is there, you can identify that up to species level. So we tried doing so, and we got a lot of success in species delimitation. We also found this, how they are phylogenetically evolved. And also we tried to understand the ancestral state. We, with the help of molecular data, we reconstructed the ancestral states. What is that? What is that? We can try to understand how those morphological characters were present in the ancestry. Like previous species, how did in the course of evolution, what was the status of that character? And not only this, how, and we also did ancestral area reconstruction, which is in which continent and during which, how many million years ago, what was the status of a particular species and how they have evolved. Everything we try to understand. That is a totally different topic. I'll discuss it some other day. But that helps us, or helped us a lot, the molecular, to point of this saying that that if you have that molecular tools with you, you can expand your horizon. You can do so many other things. Also, I will tell you about the interesting story of this polygonum. While I was doing my postdoc studies in South Korea, we came across a species called Polygonum effusus from Uttarakhand. And then we found out that these two, and there is another species from Iran called Polygonum origeolvitis. And they are morphologically very, very same. Only difference was there, the length of this peduncle was little more here. So we thought, okay, morphologically they're the same, we should merge it. But then we realized, no, we should just try checking at the molecular level whether they are same or different. But when we checked the molecular data, we found there are a lot of difference between these two species. So we did not merge them. So we have several markers, other markers also available with us. One of the most popular, I think in the last decade, this particular marker was very much popular, called RAPD, which means randomly amplified polymorphic DNA. And Vineet sir, please stop me if my time is getting up, okay? Because just let me know that if I'm taking more time. Yeah, so this particular marker called RAPD is a PCR-based technique. What we do here, we just get some particular RAPD markers. It's already available in market. What we are going to do, just arbitrarily, we are placing them in the whole isolated DNA and try to make amplification. And based on those products, like if you can see, this is species 1, this is species 2. So species 1, when we are checking with one particular this RAPD marker, it is giving us two products, product A and product B. But in sample 2, we are just giving one product. It means that particular primer is not able to amplify here. So these two are different based on these data, and then we check this on gel using electrophoresis technique, and then we can tell with surety whether these two species are same or different, or same or different. So this is how you can see this on a gel band, and this is very quick and easy, and does not involve a lot of money as well. But what happens? This has got low reproducibility. So if you just repeating it multiple times, because there's so many other factors associated with this, so it does not reproduce the same thing if you just perform it multiple times. So there is a problem. So that's why it was not much appreciated by people. And also it required high molecular weight DNA, which is not much possible in most of the cases, like unless you have fresh samples, getting high molecular weight DNA is not possible. So because of these kind of issue, this was slowly, this became outdated. What happened? But still we could produce several good work. Like if you can just go through this paper, which was published by Nagai and Chandra in 2009. They worked on Stylosanthes species, And with the help of RAPD markers, they could find out that these 4 are distinct lineages. Then we have another method called AFLP, which is a kind of advanced version of RAPD, amplified fragment length polymorphism. This method is again based on PCR, and it was developed in 1995 itself. What we do here, we are going to do, this is an advanced method and still being used. So if you want to do some complicated, if you are just trying to resolve some species complexes and all, you can make use of this method called AFLP. So what we do here, we digest the whole DNA, and then after that we ligate with some adapters. Those who are biotechnologists here, they must understand this. So do ligate some adapter, use PCR, and after that you do electrophoresis. And this is the process. You are going to just digest the DNA, and then after that you add ligate some adapters, and after that do PCR, and then you just do electrophoresis. And based on the band size differences, you can find out whether there's a species difference or there's a difference between species or not. And this method is highly reproducible. The same result, if you repeat it for many times, you will get the same result. And also, it does not require any DNA sequence information. You need not to send the DNA material to an outsource agency to get the sequencing, as we do for ITS and RBCL and other markers. And based on that band information, you can find out whether this is a new species or not. But it has also got several disadvantages because it requires more number of steps compared to RAPD. This is little more complicated, expensive, and laborious. And purity of DNA is again very important. If you want to isolate DNA from old or degraded samples, it will be difficult. And again, allelic information would be missing because it's a dominant type of marker. So, but still being used for several species complexes and all things, but for genetic mapping or population genetic analysis, this is not recommended. But using AFLP, still people are working on it, and so many good work have been already published. This is an example of eucalyptus work where they have used AFLP marker. And they have published a very good work. You know, this eucalyptus, what we see here, it is represented by almost 750 species in the world. We have several of them, but these fellows, and there is a problem between, like, if you go at the sectional level, there are a lot of complications, whether they're same species or different ones, there's a lot of confusion, a lot of problems. So these people took help of AFLP marker, and they could produce a wonderful work seeing that these are the distinct lineages and how they are different. They have correlated using more molecular data. And we are all aware of this APG. What is that? Those who are student here, plant taxonomy student, they can understand, called angiosperm phylogeny grouping as a system of classification, which was proposed long back in 1998. And using molecular data, they could find out, okay, how it is different from morphology-based classification. Here again, they use 3 different markers called RBCL and ATPB from chloroplast region, and it is 18S region from nuclear DNA. And this kept on upgrading after APG 1, 2, and 3, and this APG 4 was also proposed in 2016. You can see using these molecular data, now we know that there are 64 orders should be there, and number of families should be 416. But still, and we understand now the phylogeny of whatever are known till now, 2,95,781 taxa, how they are phylogenetically related. We know the phylogeny of it. What is remaining to understand still remaining to, so it's very less amount of taxa, kind of 183 taxa. The phylogenetic position we are still not able to understand. Maybe some other day I will explain why we are not able to understand that, because of a lot of morphological complications. So the first group is called mesangiosperm, and this is called anaclade here. And now we, as I told that the techniques are getting advanced day by day. After the sequencing techniques, Sanger sequencing techniques, we have now NGS available with us called next generation sequencing. So what we can do here, we can just sequence the whole genome or whole plastome, we call chloroplast genome, which can be helpful for phylogeny and barcoding studies. So using this genome data, it's much easy to, like you have a huge amount of data available to compare, right? Earlier, we used to compare just ITS region of that 800 base pairs we used to compare. Now you have whole genomic data to compare. So whole genome, like 100,000 base pairs now you can compare. So what we have now using these techniques, we have better resolved phylogeny. Some of the phylogenies were not, where we were not able to understand using those small regions. Now using plastome data, you can understand phylogeny in a much better way. And then also there are several problems associated with polyploidy and hybridization. Using NGS methods, you can also resolve that. And now also we have options to study species boundaries and species relationship in recent radiation. What happened? Many of the species are newly evolved. So using those smaller region of nuclear data or mitochondrial data, it was much difficult. But when you have whole chloroplast genome or whole genome data available, you can understand them in a better way. So this is how this chloroplast genome look like. We have done this in our lab as well. Then if you have this chloroplast genome available with you, what you can do, you can understand the inter and intra-specific diversity in a much better way. You can understand phylogenetic relationship. You can understand the gene composition and evolution. You can characterize the plastome. You can do DNA barcoding. You can do positive selection analysis. You can do prediction of RNA editing sites and so many other things. So using these things, there is a lady called June Wen. There is a lot of complex in Araliaceae family, and using these molecular tools, she has published a wonderful paper. And here she has used the chloroplast genomes for understanding those complexes, and she resolved those complexes which were earlier not being understood. And not only this, as we have NGS technique available, so we are using huge amount of data set. Here you can see in this the paper published by Jeffrey Sarella, and he worked on Poaceae family. And here he used almost 250 chloroplast genomes, representative of 250 different species. He used and tried to understand the phylogeny. And he again resolved several complexes which we were not able to understand earlier. In our lab also, we try to understand evolution of in OLs using chloroplast genome data. And we have published this result in PLOS ONE journal long back, Ashwini's work. And here we try to understand how they have evolved. Not only this, how we also try to establish certain potential DNA barcodes. We suggested several regions. Like if you want to identify a particular Idiocolon species, using this particular region only, you can identify your species using molecular data. And then also, there's an interesting story of two different Caparis species. This work we have produced using in collaboration with Zhejiang University in China. I had several one collaborator there. So we in Maharashtra we have one species called Caparis spinosa. As I have already described, the species which is being used in D52. In Tajikistan, my Chinese collaborator, he has collected a similar species. It's a variety of that particular species, Caparis spinosa, variety Herbacea. And there's a very few morphological differences are there. And then the difference between these two varieties are there. This particular spinosa grows at a low altitude here, and this one grows at a very high altitude. So we just wanted to understand whether at plastome level there will be any difference or not. So what we did, we sequenced the whole chloroplast genome of these two varieties and then compared them. And we found the deletion of two photosynthetic genes here. NDHF and NDHG, we found deletion of this, and this work was also published in Phytotaxa long back. So we not only compared this genomes of several Caparis taxa here and tried to understand the genome organization, and then also we found out how the LSC region or SSC region, if you go through this plastome structure, you will understand that how they vary from each other. And also, as I said, we found that deletion of two photosynthetic genes from here between these two varieties. And also, there are several genes which have been pseudogenized. And also we established DNA barcode of this particular varieties. So we have suggested several barcodes. So simply, if somebody gives you a small portion of leaf using these DNA barcode, you can easily identify. Without flower, without fruit, you can identify a species. And then not only this, nowadays, as the data availability is more, if you just go to NCBI, it's getting flooded every day. It's so much of data being submitted from across the world. Everybody is submitting data. So this recently, we all know about APG, but APG4, but these fellow using chloroplast genome data, they have tried to understand APG in a much better way. What they have done here, they have used 4,792 plastomes and 80 genes, the structure, the sequences of 80 genes, and they have tried to understand, compiling this, they have tried to understand the tree of life, or I can see this APG, the classification pattern in a much better way. And they have come up with the saying they're supporting the earlier treatment, but some of the phylogenetic where there is a problem, they try to understand in a much better way. Not only this, these molecular tools also help you to understand the population level variations. Some of the morphology-based taxonomy, we simply leave some of the things saying that, okay, it's just population level variation because we go to different population or different, because of some ecological conditions, they are showing some morphological diversity, morphological differences are there. Nowadays we have several techniques available. One of them is called RAD-seq, which is a restricted site associated DNA sequencing. Another one is called GBS, genotyping by sequencing. And using this, you can try to very well understand how much molecular differences are there between multiple different individuals of one species. So there's a wonderful paper on coffee. We all know coffee. And we might be knowing that coffee actually, this is known by more than 120 species in the world. So these fellows, and the phylogeny of the coffee was not very well understood. So these fellows, they took help of this GBS technique and tried to understand the phylogeny of this coffee. And they produced this phylogenetic tree and saying that actually the coffee, what we can see, they are divided into two different plates. Means one, there are two different groups of coffee. One group, they don't have any caffeine content. And there are very few, this all group, you can see they're all caffeine free, and which they named as the Iucn. And another clade was there, where they had presence of caffeine. And most of them are actually found in Madagascar. And they tried to understand not only the phylogeny, but also tried to understand how biogeographically, their biogeography, how they have been distributed in different continents, how they have evolved in different continents using molecular tools. Not only this, they try to understand the evolution of caffeine content in different species of coffee. So the line where you can see this red color is there, where they found the most highest amount of caffeine was present. And here you can see a bluish line. And this, maybe I will explain you some other day. I don't have much time right now to explain this tree, but you can very well understand how a particular chemical compound has also evolved during the whole evolutionary history of a particular genus or a family. And maybe I will recommend you to go through this paper and you will understand in a much better way. And this paper also is a wonderful paper which was published in Systematic Biology, having impact factor of 10. And what they have done, there's a cryptic species complex was there in a genus called Viburnum. We also have that particular genus in Arunachal Pradesh. So using this restricted site associated DNA method, RAD-seq method, they tried to understand 3 different cryptic Viburnum species. There were a lot of fighting among taxonomists saying that some of them saying these 3 are 3 different species. Some of them will say, no, they're the same. So there was a lot of fighting going on. So this Donohue et al., they and these fellow Sticks et al. From Donohue lab, they performed a wonderful study. And they, with the help of molecular data, RAD-seq data, they could finally prove it that these three are not species, resolve this species complexes, and they could prove out how these 3 are different at a molecular level. And this was published in a very good journal, Systematic Biology. And also wonderful analysis they have provided. using the same, and you can see three distinct lineages are there, three distinct ways. There's also a technique available nowadays with us called genome skimming. As we know that if you want to sequence the whole genome, that would be quite expensive. So there is a method called genome skimming, which at a much cheaper rate you can do. What it does, it simply does a kind of shallow sequencing, like take some region of that particular whole genome and then try to find out the difference between the sequences. And you doing this method, you try to produce the phylogenomics or phylogeny of a particular plant group. This method has also been proved very successful, and so many papers nowadays you can find where they have used genome skimming method, and they have resolved so many species complexes as well. Many of us, we feel that what taxonomy, when we are doing, it's not a fashionable science, it's a very old fashionable science. And there have been several papers, but believe me, taxonomy is not an old fashioned science. If you can see, if you talk about impact factor, many of this is okay. If you're doing taxonomy, you will not be able to publish good and high impact papers. But you see all these examples. This was published in an impact factor 10. This was also having impact factor 4, 2.7, plus 1 molecular phylogenetic evolution. They have done nothing but just it's plant taxonomy only. So only thing they have used some other some advanced tools and technique to do the same thing what you're doing using morphological data. And also nowadays, since we have APG4 already, we know. So as we are getting much and much data from across the world, so people who are working in US, who are working in Europe, who are working in India, now we have a common hub, and you can simply share the sequences and you can do your own analysis. So after APG4 was proposed, after that, after 2016, so many new things also have been added. Like now we know a new family called Tiganophytaceae in brassicales, we know using, and that also was established using molecular data. And there's a new family also, Mistropetalaceae in Santalales. And then so many new additions are there, so many changes are there. So we are very sure that APG5 is also soon going to come. And as you can see, this brassicales Tiganophytaceae, I've just shown you this example. So altogether, the take-home message is what? Although we say that molecular phylogeny is a great improvement to earlier system, but it is not definite answer. This is a continuously evolving system. What are the tools we are using presently? Maybe if tomorrow, if we see some new tools will come, using which we can do a much better taxonomy. Right? So it's a continuously evolving system. It's not like a checkpoint. Then molecular systematics has also its limitations with recurring convergent evolutions. What is that? Already we have seen the case of Euphorbia and that cactus based on these kind of characters called homoplacious character, they are very much deceptive sometimes. So they also, the molecular data, sometimes we get deceived if it is somehow by the homoplacious characters. So Stevens has written a wonderful thing. So those who are actually doing morphology-based taxonomy, please don't get disappointed. And those who are doing a small work, also kind of small revisionary work of particular region, flora of a particular region, you are adding a lot to science. So don't get disappointed that nowadays so many new things have come up. I have to just cope up with all those things, not able to do that. Don't think like that. You are also adding a lot to science. So that's why I would like to quote Mr. Stevens, that he has written, our morphological, anatomical and chemical knowledge of many critical taxa is woefully complete, particularly in tropical regions like countries like India, Cambodia, Vietnam. There's so many groups which we are still not being able to explore properly. So still and much basic and unfortunately perhaps unfashionable work must be undertaken to clarify the distribution of morphological, anatomical, and chemical characters. So whatever you're doing, keep on doing that don't get disappointed saying that I'm not using molecular tools, so my science is very old-fashioned one. No, it's not like that. You are also adding a lot. You make a wall brick by brick, right? That's why whatever you're doing, keep on doing that. If you're getting opportunity to use molecular tools, well and good, you do that. And definitely there is, in the future, there's a need to combine of molecular data with new morphological data for a better understanding of the evolution of angiosperm biodiversity. With this, I would like to conclude with the quote. For most of the things, we need to update ourselves. Update is very much important. Whatever you're doing, keep on reading more and more. What are the new things happening? But you keep in mind that molecular taxonomy, if I'm saying, I'm not talking about a different branch. It's not a substitute to classical taxonomy. Classical taxonomy, the baseline. So even if you are doing molecular taxonomy, to analyze the data, you must have, you should have the understanding of morphological diversity of the morphology first. Then only you will be able to do molecular taxonomy. So it's a complementary to it, right? So recent taxonomic papers, if you see, which includes DNA information alongside morphological information. It's not like simply based on molecular data. You will see that they discuss a lot about morphology. So without DNA taxonomy having replaced the traditional taxonomy. But many of you will ask, is it the time, in a Rao sir actually is very famous taxonomist, very senior one, and he in his paper he has written, is it the time to be a chemo, cyto, histo, eco, molecular taxonomist? My point, I say yes, it is the time to become like that, because unless we compile all the data, you'll not get a very clear picture. With this cartoon, where the two frogs are talking to each other, looks are not everything. It is what inside you that really matters. A biology teacher told me like that. With this, I would like to thank to all my lab members, and also to my director of BSI, Dr. Mao, and also to Dr. V.S. Rawat, who invited me, Dr. V.K. Rawat, who actually invited me for this workshop and gave me a chance to interact with you all. Thank you so much for your patience listening. I'll be happy to take questions. Thank you so much.
Thanks, Dr. Ritesh ji, for wonderful lecture, and you have spared your time for this session. And I hope all the offline, online candidates will be benefited from this excellent and informative lectures. Thank you. Thanks a lot.
Thank you so much.
And for your team and for your kind words.
Thank you so much, sir. I would like to have, I'll be happy if someone asks questions, particularly the students.
If the students, if they have any doubts, any question, please feel free to ask our sir.
Good afternoon, Dr. Sir.
Good afternoon. Yeah. How are you, sir?
I'm fine. Hope you are also fine, sir.
Yes. I'm good.
Thank you very much for this extraordinary presentation. It was very good, brief and concise. Actually, I have had a few rare opportunities to listen to your presentation on plant molecular studies, and it was always mesmerizing and captivating for me. So you also mesmerized me with your presentation. So thank you very much. Just I want to know a little bit about one thing. Specifically after the advent of this angiosperm phylogeny group, across there has been a lot of work on molecular molecular studies, which has been on continuous rise. And off and on there has also been a discussion that this Herbarium specimens, they should be arranged as per the angiosperm phylogeny grouping systems. Yes. I just want to ask one question, Dr. Saab, what is the status of these molecular studies in India? Whether there has been any particular family or genus which has already been completed, holistically completed, so that you can now say that yes, molecular studies on this group of plant has already been completed from India. And accordingly, at least we can prepare a database that yes, since it is complete, now we can arrange this type of plant groups in our herbarium. So what is the status in actually? Let's just throw a light on that, please.
Yes. Thank you. It's a wonderful question, and I was expecting it indeed. So yes, actually, it's all about research, and it's a kind of continuous process. You cannot call it complete, right? But still, we are putting a lot of effort to understand, as I told, understand it bit by bit. As I shown you this idiopathic. It was very complicated and it takes a lot of time. So unless we have multiple groups working in India, Dr. Pandey sir is there already. His group has already worked a lot in molecular taxonomy, and they have also completed a lot of work, a lot of genera they have taken, a lot of species there, a lot of species complexes are there, which they have resolved. But that's not enough. India, the biodiversity is so big, it's huge, right? So many, particularly for the tropical countries like Vietnam, Cambodia, Laos, India, Sri Lanka, and all these countries, we have a lot of biodiversity, but still infrastructure-wise or resource-wise, we are kind of not sufficient enough. So we have very few labs where we are doing molecular-based studies. So though in BSI also has started, and now in CNH, that a new molecular lab has been started. So there are not many groups which have already been worked out. Like I would say in my case, we have completed work on Idiocola. Out of 400 species which are found in the world, we know the phylogenetic position of those small group, like 1/4 of that particular group. But unless we have data from across the globe, I cannot call that study complete. So unless we have sequences of all those 400 species, we cannot, unless we compare them together, putting everything together, it will be called as incomplete only. In Caparis, at some level, we could do that because we collaborated with people from South America. We collaborated with people from Korea, who provided us samples of multiple countries as well. So it's in review now, and very soon you will find. And that study, we would like to say that at some level we have tried to complete it from our side. Thank you so much.
Thank you very much, Dr. Saab. Just is there any primitive group on which actually this molecular systematic study has been done from India, belonging to Magnolids group?
Magnolids, I think people are doing from India. I don't think so. Some primitive group they have worked. Like now, primitive, which one you say? Suppose if I'm talking about anaclid, right? So nymphias also not been worked. Ostrobellus is not there. So in that particular, like which actually that fall under MC group, many of the species families, they have not been worked out from India.
Thank you very much for your insight, giving this insight, Dr. Saab.
Thank you.
Thank you, sir, once again for your wonderful presentation. Now, I will request our Rawat sir for briefing our next speaker.
Murugan sir.
Murugan sir.
Sir, good afternoon, sir. Good afternoon, sir.
Sir, you are on mute mode. Yeah, please unmute yourself. And good afternoon from Pune.
Good afternoon,
nice
sir.
to see you online, sir.
Okay.
Good afternoon, sir.
Good afternoon.
Now, just I'm going to introduce Murugan sir, and he's going to deliver a lecture on phytodiversity of Southern Western Ghat. Presently, Murugan sir is the scientist, joint director and head of office in headquarters BSI. He has done his MSc and PhD from Tirunelveli, and till date he has described 44 species and new distribution records for India 38 species, Tamil Nadu 24 species, Kerala 1 species, Andhra Pradesh 3 species, Andaman Nicobar 9 species, and he has also discovered 19 species. He has done also one species nomenclature change. And his PhD topic was taxonomic studies on the flora of Tirunelveli Valley Hills, polypetally under the guidance of late Dr. B.S. Manikam sir. And he has published 4 books, 3 book chapters, and 124 research articles. And till date, he has completed 10 research projects and participated in 7 workshops and trainings and 11 conferences and seminars. He is the member of 4 reputed societies like Indian Association for Angiosperm Taxonomy, Indian Science Congress Association India. He is the life member, Andaman Science Association India, he is the life member, and National Environment Science Academy. He is the fellow of 2 societies, fellow of Indian Angiosperm Plant Taxonomy, fellow of Journal of Andaman Science Association. And he is very knowledgeable person and very kind and gentleman. And now he's going to present about phytodiversity of Southern Western Ghats with this special reference to Tamil Nadu. Thank you, sir. Please proceed, sir.
Thank you, Dr. Vidit. Very good afternoon to all. On behalf of Botanical Survey of India, I am representing to deliver this lecture on biodiversity of Southern Western Ghats. But I made small change in my title as with special reference to the Tamil Nadu. First of all, I thank you, the Botanical Survey of India, in collaboration with Arunachal Pradesh Science and Council Technology to conduct this capacity building program, plant taxonomy, systematics, and conservation. So this is a nice step Dr. Vinitra has taken to disseminate the scientific knowledge to the students community. So now I will come to my title, Biodiversity of Southern Western Ghats with special reference to the Tamil Nadu. Before start my lecture, no earth, no life. Because in earth, providing water, providing shelter to the humans. So now today trend is there is a lot of disturbances are due to the human beings or any other natural factor. So to protect the plant wealth on earth, United Nations in 2018 developed the program for conservation of earth in the form of protected areas from 244 countries and territories covering more than 46 million square kilometers. These protected areas are distributed across 11 CPD regions, namely Africa, Asia, Caribbean, Central America, European, Middle East. Sir, excuse me, I am audible, sir.
Sir,
Hello?
you are audible, sir. You are audible, sir.
Yes, sir.
And slides are visible, sir.
South America, Southern Ocean, and the areas beyond national jurisdiction. So this green color indicates up to 2014, what are the protected areas available on Earth. This red color indicates since 2014, these are the new protected areas developed by the United Nations. So when you come to India, we have total 981 protected areas comprising community reserves, conservation reserves, wildlife sanctuaries, national park. It includes 1,71,921 square kilometers in the total area of India, of which only we are having 5.303% of protected areas in India. This map indicates what are the protected areas available in India, the red color, all that Andaman Nicobar, Tamil Nadu, Kerala, Goa, Gujarat, that this indicates what are the protected areas available in Indian regions. So, India, one of the 2 mega diversity centers in the world. India has 3 mega diversity centers, namely Himalaya, Western Ghats, and Eastern Ghats. Among the 3, the Western Ghats is running on the west coast of India, about 1,500 to 1,600 kilometers long. It covers from various states like Gujarat, Maharashtra, Goa, Karnataka, Kerala, and Tamil Nadu. This Western Ghats is home for more than 50 million people and also source for water for entire peninsula India. This Western Ghats influences both the monsoons, namely northwest monsoon, southwest monsoon. And also it retains only 7% of the original forest cover. This Western Ghats is great older than the Himalaya. It occupied 1,40,000 square kilometers, that is centered by only 30 kilometers pocket at 11 degree north. The Western Ghats contain exceptional levels of plant, animal diversity and endemism. In particular, the level of endemism is about 5,000 plant species, out of which 650 tree species found in the Western Ghats, of which 352 are endemic to Western Ghats and also India. And also animal diversity is also so exceptional. According to Agamdalo and Nair, this Western Ghats classified into 3 phytogeographical zones, namely Northern Western Ghats, Central Western Ghats, and Southern Western Ghats. It includes Malabar, Nilgiris, Ornamental, Palni Hills, Tirunelveli Hills Complex, and Trongo. The plantation of Southern Western Ghats estimated over 4,500 flowering plants, of which 1,500 endemic to Western Ghats. Out of this, 15 genus are very common in Western Ghats, namely Iucn Categories, Silent Vallia, Jaunegia, Jardodia, Pyolopis, Phytoconthus, Poeciloneron, Adenone, Metrometrus, Botonophilium, Pseudoclecidian, Panacesua, Octotropis, Pseudodia. When it comes to Southern Western Ghats, there are 5 microendemic centers, namely Shimoga and Canara with 12%, Agastyamalai Hills with 38%, Anamalai Kai ranges with 12%, Palli Hills with 8%, Nilgiri, Salem Valley, Wayanad, Kodagu region with 30%. Among the Western Ghats, the southernmost state in India is located between 11 degree 7 minute 37 second north latitude and 78 degree 39 minute 24 second east latitude. It covers an area of 13,58,000 square kilometer, composing 4% of the total geographical area of the country, with the elevation ranging from sea level to 2,637 meters sea level. Topographically, the state is divided into Western Ghats, Eastern Ghats, Central Plateau, and coastal plains. The average rainfall varies from 900 to 3,000 millimeters, being benefited by both southwest and northwest monsoon. The state enjoys tropical humid climate and has varied topography and habitats. The Western Ghats of Tamil Nadu runs from Kanyakumari through Tirunelveli, Tenkasi, Virudhunagar, Theni, Tindikal, Tirupur, Coimbatore, Nilgiris, and Erode districts. The highest peak in the Western Ghats of Tamil Nadu is 2,637 meters sea level, and the Kolaripata with 2,629 meters sea level, located in the Nilgiris, Vandara peak in Palni Hills. The Western Ghats of Tamil Nadu harbor 4 microendemic centers, namely Agastyamalai Hills, Palni Hills, Anamalai Hills, Nilgiri, Wayanad, Kodagu, 5 junctions. In addition, that there are very important hills like Mahendragiri Hills, Varsanad and Aldipatti Hills, Cardamom Hills, come to the vegetation of Tamil Nadu, there are 7 types of vegetation are available. The scrub or thorny forest, which is adjacent to the foothills of Western Ghats. Above scrub forest, you can find the dry deciduous forest. There you can see that animals like panther, cheetah, you can find that area. Above dry deciduous forest, you can find that moist deciduous forest. That is nothing but a mixing of dry deciduous forest with the evergreen elements. So that is called a moist deciduous forest. Then forest, evergreen forest, tropical montane evergreen forest, that is called a swallow forest. That will be spread across the high altitude grassland. Then coming to grassland, there are 2 categories are there: high altitude and low altitude. Below 800 meter sea level, that type of grassland called low altitude grassland. Above 800 meter, that region called high altitude grasslands. But the grass elements are typically vary from high altitude grassland and low altitude grassland. In low altitude grassland, you can see a lot of diversity of grasses than high altitude grassland. So in Tamil Nadu, we have 4 major type of protected areas are available: biosphere reserve, national park, tiger reserve, wildlife sanctuary. There are 3 biosphere, namely Nilgiri Biosphere Reserve. Representative area of Western Ghats covers 5,670 square kilometers. It was set up in 1986 as the first biosphere reserve in India, borrowing the 3 states, namely Tamil Nadu, Kerala, Karnataka. And also more than 3,238 plant species, including 197 narrow endemic plants are available. And we can come across 684 vertebrate species have been registered. Several hundred plant species have been used for medicinal, industrial, or food uses. Next is the Agassimalai Biosphere Reserve, located in the southernmost end of the Western Ghats. It is spread across the 3 districts of Tirunelveli, Kanyakumari in Tamil Nadu, and Thiruvananthapuram, Kollam, Pathanamthitta district in Kerala state. Of the total reported area is 5,432 square kilometers, of which 2,560 kilometers is reported from Tamil Nadu, 2,871 kilometers reported from Kerala. This region is also home of Kani tribal, the Dravidian tribe. The concentration of endemic species is much richer in this center when compared to the other centers of endemism in Western Ghats. The peaks reaching 1,868 meters above sea level consists mostly of tropical forests. This site is home to 2,254 species of higher plants, including about 400 that are endemic to these regions. Next is the Gulf of Mannar spider that is coming into the coastal regions. So we have 3 national parks. Indira Gandhi National Park, Coimbatore, it was declared in 89 with the area 84,149 hectare area. Next, Mudumalai National Park, located in Nilgiri district. It was declared in 1940, 21,776 hectare area, followed by Muhurti National Park, that also located in Nilgiri district. It was declared in 2001, the area with 7,846 hectare area. Then, when come to the tiger reserve, there are 4 tiger reserves are there. One is Kalakada Mundandurai, that is the first tiger reserve in Tamil Nadu state, established in 1988, which is located in Tirunelveli, Kanyakumari district of Tamil Nadu, with 89,500 hectare area. Followed by 2 tiger reserves declared in 2007 as Ornamental Tiger Reserve, Mudumalai Tiger Reserve. Then recently, 2021, Government of India declared Seliputtur Megamalai Tiger Reserve with 1,657 hectare area as a tiger reserve. Tamil Nadu has 9 wildlife sanctuary, namely Indira Gandhi Wildlife Sanctuary located in Coimbatore, Kalakadu Wildlife Sanctuary in Tirunelveli district, Kanyakumari Wildlife Sanctuary, Kanyakumari, Kodaikanal Wildlife Sanctuary, Tindikal, Megamalai Wildlife Sanctuary, Theni, Mudumalai Wildlife Sanctuary, Nilgiri district, Mundandurai Wildlife Sanctuary, Tirunelveli district, Nellai Wildlife Sanctuary, Tengasi and Tirunelveli, Sriliputur Wildlife Sanctuary. This all area well documented by the Botanical Survey of India. Some very interesting area in the Western Ghats parts of Tamil Nadu is the highest peak, that is Tottapatta in Nilgiri district with a 2,637 meters sea level. Kolaripata peak in Nilgiri district with a 2,629 meters sea level. Vandara Rao Peak in Palni Hills with a 2,553 meters sea level. Kadikadu Peak in Nilgiri district with 2,589 meters sea level. When come to the botanical exploration, several botanists are done wonderful research and documented the flora of India during the British period. Robert White made extensive survey in the Western Ghats of Tamil Nadu. He described as a taxonomy about 110 new genera, 1,067 new species from flowering plants. And also he contributed Icon Splendor of India Ornamental, Illustration of Indian Botany, Spicigilium nilgerans. Followed by Simit made extensive survey in Coimbatore and Nilgiris and published as a flora of Coimbatore and the Nilgiris. Followed by Kokkonekar, surveyed the Karnataka regions in the name of Kannada and neighboring areas of Maharashtra and Malabar. After that, R.H. Pedum done wonderful work in the Western Ghats of Tamil Nadu and published as the plants of British India, plants of Southern India, followed by James Kite Campbell. He has made a wonderful survey with team. They published the flora of Princess of Madras from 1847 to 1925. After that, T.F. Odilon made a wonderful research in forest of Trangur. Then he publishes Forest Trees of Trangur. Followed by Faison made extensive survey in Nilgiri and Palni Hills. He published as flora of Nilgiri Hills and flora of Southern Indian Hill Station. After that, Ramarao made extensive survey in the Trangur Hills and published as flowering plants of Trangur area. After that, Fisher he made a wonderful survey in Anamalai Hills and publishes in the name of the flora of the Anamalai Hills, Coimbatore district, Madras. Followed by several eminent people like C.A. Barber, M.A. Lawson, K.C. Jacob, J.L. Ellis, Subramaniam, P.D. Sarma, A.G. Borni, C.C. Galdar, M.S. Ramaswamy, J.J. Sosap, A. Milgold. They are the pioneer plant collectors of Southern Western Ghats of Tamil Nadu, are the main reason for the richness of the Madras herbarium today. These are some important publications from the Western Ghats region. Flora of Tamil Nadu by Dr. Ian Henry and all. They published 3 volumes. Then Nair and Chandrapose published flora of Coimbatore. Then our former director M.B. Nair and K.K. Nair published flora of Coimbatore, Kuttalam in 1986. P.D. Sarma, our former director Dr. B.D. Sarma and group published studies on the flora of Nilgiris, Tamil Nadu. Then Dr. Joseph published Orchids of Nilgiris. Recently, our eminent scientist Dr. V.J. Nair and Dr. Altaf Kabir published flora of Tamil Nadu grasses. Then Dr. Monikam and their team published flora of Tirunelveli Hills. Based on the intensive and extensive survey by the Botanical Survey of India scientists, so far they have discovered 8 new genera and 200 new species, which are discovered by the scientists of Coimbatore. When come to the phytotypes of Tamil Nadu, we have 1,077 species of fungi and 5,555 lichen species, 1,263 algal species, 712 parasitic species, 275 developed species, 4 native species, and 6 exotic species of gymnosperm. Overall, the angiosperm has more species diversity with 6,723 taxa. That is the #1 state of India regarding the plant diversity. This is nothing but that phytoplankton of Tamil Nadu, we are comparing with the angiosperm with 71%, fungi with 11%, lichen with 6%, bryophytes with 8%, pteridophytes with 3%, gymnosperm with 1%. When come to analysis of the Western Ghats of states, Tamil Nadu in the top in position with 6,723 species, followed by Kerala with 5,094 species, Karnataka with 4,500 species, and Maharashtra with 3,134 species. When compare the endemism in Western Ghats, when compare the endemic species, among the all four states, Tamil Nadu is top one with 410 species, followed by Kerala with 357, then Maharashtra with 278 taxa, and Karnataka with 130 species. This slide indicates a number of species available in the various states. When compared to Tamil Nadu to Tripura level, Tamil Nadu has more species with 6,723. Then some of the states are not included due to the non-availability of some literature. Tripura with 1,462. This is the bar diagram of the velocity diverse in the various states compared with Tamil Nadu. When come to the endemic genera in southern Western Ghats, already I told you during that speaking about the Western Ghats plant diversity, Iucn Henry I wrote a report. This genus endemic to Agastya Malai Hills. So it named after our former BSc scientist, Dr. Iucn Henry. So the name as Iucn Henry. Henrya rotundifol, about the Piolepis nervosa, that is belongs to the family Apocynaceae. This is a very useful plant species which is restricted to Neilgiri Basperiosum of Ooty. Then the genus called Triatoconus, it has 2 species. One is Albiflorus, another is Piolepis. This genus endemic to Western Ghats from the Tamil Nadu and Kerala states, followed by Jardonia indica belongs to Jardoniaceae. It has only 1 species which is endemic to Western Ghats. Then this is Metrometer vinodus, very interesting plant species. After a time, recently it was discovered from the Kerala part of the Western Ghats. This genus also endemic to Tamil Nadu and Kerala states, followed by Pseudoclerchidia. This genus also endemic to Annamalai Tigers. Then this very interesting composite number called Adenone indicum. This genus is endemic to India, but the species is distributed in Western Ghats of Maharashtra as well as Tamil Nadu. Then Octotropis tramorica, this plant belongs to the family Rubiaceae. It is an evergreen element, very common in the Agastya Malai Hills in both Tamil Nadu and Kerala parts of the southern Western Ghats. Otonepinus stipulosus belongs to the family Serpulaceae. It is endemic to southern Western Ghats. Followed by the genus called Pseudodia speciosa, belongs to the family Rubiaceae. That also common in southern Western Ghats. This crass genus called Silent Valley. I think in the student assembly, everybody know that Silent Valley National Park. So in honor this Silent Valley National Park, they named as Silent Valley area. Then another Vanassusua pedata belongs to the family Apiaceae. Then everybody knows that Parsaminaceae is a very good family for with attractive flower. For example, the genus called Impatiens has a lot of attractive ornamental wild plant species. Impatiens orchioides, Impatiens leptatura, Impatiens levingii, Impatiens tangachi, invasive latifolia, invasive latica. These all species endemic to Western Ghats of Tamil Nadu. Then some of the orchids are home for Western Ghats, namely Pulpophyllum aureum. It is epiphytic orchid, very common in the Agasthyamala hills. Then Chylostis is the glandulosa. It is a epiphytic orchid found in the Nilgiri hills. Then Coelochyni mosaic, that is very occasional. Now the population also decreased due to the habitat and man-made disturbances. Then this is another epiphytic orchid called Dendrobium anamalayanum. That is a very rare in Meghamalai Wildlife Sanctuary. Dendrobium hainianum. This is a epiphytic orchid found in the Nilgiri Biosphere Reserve. This Protoceras holtami. This species is described from the Vellingiri Hills. But we have some doubtfulness. This species may be then come to the Melastomaceae. There are several genus are Memisilon, Melastoma, Oprodia, then Sonarella. This genus, Herbaceous genus called Sonarella, has very good ornamental value like Sonarella tenella, then Sonarella speciosa, Sonarella vesicolor, Sonarella grandiflora, that is endemic to Meghamalai Hills, Sonarella Parameswarani, that is nothing but Parameswarani, our former director MB Parameswaran Nair. To honor him, BSA scientist named Sonarella Parameswarani. Then Sonarella Palni, this is from Palni Hills. This is endemic to Kodaikanal Wildlife Sanctuary. When come to the Rubiaceae, there are very interesting plants are found in the Western Ghats of Tamil Nadu. This Hidiotis paxifolia found in the high altitude grassland in the fringes of the Sola forest. Then Hidiotis purpurosum, that is very common species in high altitude grassland and also fringes of the Sola forest. This Hidiotis nayeri, it also found in the high altitude grassland in the Vellingiri Hills of the Tamil Nadu region. Then Lasiantha cinereus, very common species in the Western Ghats of Tamil Nadu, belongs to the family Rubiaceae. In Ooty, Nilgiri Biosphere, this Ophioryza pigarensis, that is a rare species recently discovered by our BSA scientist. Actually, this Psychotia cyanuriana belongs to the family Rubiaceae. It was collected and identified as Psychotia octosulcata. Based on the critical studies, our BSA scientist named as the plant selected by Dr. Iucn Henry to honor him, so we named as Psychotia henryana. Recently, one of the students from Botanical Survey of India collected this plant from Western Ghats of Kerala. So with the fruits. While describing this species, it was collected with only flowers. Then the Acaciaceae has a lot of interesting genus like Strobilanthes. If you know that this Strobilanthes, once the flowering completed, fruit will set after the plant will die. Like this, the variation will be, the life of the Strobilanthes species will be 7 to 12 years. Like the Sfoliosa, Strobilanthes catromella, Strobilanthes perotitiana, Strobilanthes amabilis, Strobilanthes virendra kumariana, Strobilanthes homotropa. These are the species endemic to Western Ghats of Tamil Nadu regions. Recently, our Botanical Survey of India initiated new postdoctoral research program as Acharya Sagri's postdoctoral research program by the Botanical Survey of India. So one of the students from Coimbatore region, he studied on the narrow endemic species of Nilgiri Biosphere. He documented and collected and described as new species Dipkadi Coimbatore from this Sirivadi Hills area. Then Eugenia bolomatiana from Polamati Hills of Coimbatore district. Then Eugenia vellingeriana from Vellingiri Hills of Nilgiri Biosphere. Then Sonarella Coimbatore from Ornamental Tiger Reserve. Then Dendrobium gobalani from Southern Western Ghats by Mr. Sulaima. The Kainato niligrensis by Murugan and Ravichandran. This species done by Dr. Ravichandran during that PDF program. Then also he has discovered a lot of new species like a Fabernaria polyoda, Fabernaria richardiana, Penium nodum from Nilgiri Pasperisov, Limbacean laticornis per Nilgiri Pasperov. Then Dr. Armam of Botanical Survey of India, Coimbatore, he recollected this species after type supported from Megamalay Wildlife Sanctuary. Then Iucn Nilgiri rediscovered from the Nilgiri Pasperisov. So when compare the species and generic diversity, this family Fabaceae has more diversity. Then followed by Poaceae, Asteraceae, Rubiaceae, Orchidaceae, Cyperaceae, Acanthaceae, Euphorbiaceae, Lamiaceae. When come to the endemic species with the family, among the all endemism species, Parsaminosae has more endemism with 98%, followed by Melastomentaceae with 57%, Acanthaceae with 47%. When study the OT endemic species, Memicelon has more number of species with a 16 number, then followed by Lixia with a 15 number, Sibylophus, Cinnamomum, Syzygium, Actinodaphnine, Cloacadia, Crevia. Then when study the endemic herbaceous elements, the genus Imbaceans has more species, followed by Eriocolon, Fimbrisalis, then Sonarilla, Auspicia, Piper. Then based on the recent extensive survey and literature survey, we found that these are the genus having description of more new species like Imbaceans, Eriocolon, Sonarilla, Dimeria, Memicelon, Syzygium, and Tripoga, Fimbrisalis. Hello, Dr. Vineet.
Yes, sir. Yes, sir. I am here, sir.
So, based on my presentation, I summarize that among the all Indian states, Tamil Nadu has more species diversity with endemism. So I will conclude my presentation, Dr. Vineet. So if the time is over, if you have any doubt, you can ask.
Okay, sir. Thank you, sir, for your insightful and wonderful presentation. And I would like to discuss here that sir is the head of office in headquarters. So in spite of his busy schedule, he has accepted our request and he has prepared this presentation and he has given his valuable time for us. So students will be benefited for this beautiful presentation, sir. Thank you. So much thanks from our center and APSC, all the staffs. Thank you, sir. Thanks a lot, sir.
Thank you, Dr. Vineet, for a wonderful opportunity for me to showcase my plant wealth of Tamil Nadu.
Yes, sir. बहुत beautiful presentation था, sir. और आपने एक्सेप्ट की हमारा जो इनविटेशन और आप इतना रेस्पॉन्सिबिलिटी है सर के पास कि मतलब पूरा रीजनल सेंटर्स का मैंने रिक्वेस्ट किया था कि सर आप प्लीज आधा घंटा हमें दे दीजिए तो सर ने दिया और विवेक सर
thank you very much, sir, for accepting our invitation.
Okay, thank you, sir. Thank you, sir.
Okay. Also invited for the
And sir,
thank you, Dr.
One request is there, sir.
Thank you, Dr.
In future,
sir.
we want a helping hand from your side.
Thank you. Now it is lunch session, so all the offline candidates are requested to go for lunch and we will assemble after 45 minutes. Thank you, sir. Now session is over. And sir, Morgan sir, you are requested to deliver your blessings for us in benedictive function that will be held after 1 hour, 1.5 hour. Sir, please request.
At what time, Dr. Sam? At what time?
Now it is 1:50, sir. 2:30, around 2:30 it will start, sir.
Okay.
Thank you, sir. Thank you, sir.
Thank you. Okay.
Jai Hind, sir.
Jai Hind.